Okay na, Ma'am Ler. Nakita ko na.
Ma'am Sonia.
Hello, JV?
Hello, ma'am. After ko po dun sa how to get a certificate of participation, kayo na po yung next mo.
Okay. Sige.
Okay po. And then, Ma'am Larry, may recap pong nakalagay. So, tama po ba na recap ang ilagay ko? Ma'am Larry? Ma'am Larry?
Yes, oo. Sorry.
Okay po.
Mag-share ako ng PowerPoint pag nag-recap.
Okay po.
Thank you.
Mary, hello po, good morning po. Mary?
Yes.
Yung regarding po dun sa streaming po, gagamitin po natin yung account niyo po or yung PSDB official po.
Oo nga. Bakit hindi siya mag-connect doon sa PSDB official?
Ma'am Larry?
Ma'am, parang okay na po.
Yes, oo. Sorry.
Okay po.
Mag-share ako ng Ay, sorry. Wait, ako yun. Okay na ba? Ano na ba sa PSDB? Or sa ano ko pa rin?
Sa PSDB na
Sa
po.
streaming po, gagamitin po natin yung Okay. Thank you, Josh. Thank you po, Ma'am Larry.
Wala pa dyan. Wala. So, teka po, dyan po sa poster presentation, may mag-a-announce pa po, sino pong mag-ano dyan, si Sir Ahmad? Tapos magpa-play lang ako.
Ako na po, ma'am, yung magtatawag po sa inyo.
Okay.
Okay po. So, pag in-announce ko na po yung 3 best posters, yun po yung time na
Okay.
ano po.
Pero wala pang result, di ba?
Yes po.
Okay.
Ma'am Larry,
Yes po.
iimikin ko lang po ba yung name or
Ano daw?
iimikin ko lang po yung name, no? Hindi na po ako magpo-provide ng slide nung tatlong nanalo. Anong best posters?
Yung sa best posters. Walang binigay sina poster ko, may.
Sige po, pinikin ko na lang po yung name.
May slide ka naman ng winners. Ganun na lang.
Wala po, ma'am.
Ay, wala pala. Hindi pa pala winners. Ano lang pala yun.
Best posters pa lang po. Pero hindi ko po ilalagay yung name nila. Like, babanggitin ko lang po.
Baka pwede yung i-flash na lang yung digital nila na poster. Pero baka matagalan tayo or mas complicated na kukunin pa, hanapin pa yung digital poster tapos i-paste dito.
Ma'am. Medyo mahirap na din especially medyo malaking trabaho din siya.
Oo.
So, imikin ko na lang po.
Tapos acknowledge na lang kung nandoon na sila. Parang itanong mo lang as the session moderator, or do we have the representative or the author?
Okay po.
Sino tatanungin, si Sir Ahmad, no? Baka meron na. Tatanungin natin.
Ayun. After po ng poster presentation, mali po pala yung hindi ko pa siya na-edit. Poster live Q&A na po. So, tawagin ko po yung best speakers and then first po na poster presenter yung tatanungin din, second and third. 5 minutes po per poster presenter po. At saka thank you for participation and then
paano pag hindi sila umattend? Ngayon yung winner.
Parang i-inform po yata ni Sir Ahmad yung tatlo. Or hindi ko po alam kung Sir Ahmad. Tapos ilagay ko po yung slide na please be back at 1 p.m. for session 5. And then yun. Yeah, tapos na po. Malinaw po ba yung audio ko?
Clear naman, JV.
Po.
Ay, nandito na po si Ma'am Andrea. Papasok na po siya. Pa-check na lang po yung kanyang audio and share screen with audio. Good morning, Miss Andrea.
Morning ka, morning. Oo, basta sa malay pagkukap sa sulod.
Hello? Hello, Miss Andrea? Oh, can't unmute. Please. Ay, ako pala yung host.
Hello?
Okay, then.
Kailangan ko rin bang i-check yung screen?
Yes, please. While you're sharing your screen with the audio and then, okay lang ba na meron kang video din while talking?
Yep.
There you
Yep. Ay, nilipat ako ng room. So,
Nasa lab ka ba?
oo. Yun. Wait, check ko lang yung screen. Paano to? Kita ba? Or dapat i-swap screen ko? Tama ba?
Kita, okay.
Yun yung tamang... Kasi
Let's go.
meron ako nung notes. Oo.
And if you want to
Okay.
use pala yung Zoom background, we put it here in the chat box if you want to use it. Yung background nung ginagamit namin ngayon. Pero hindi naman necessary depending din sa capability nung Do you
Oo,
have-
hindi ko alam kung...
Yes,
Sorry?
go ahead.
Hindi ko alam kung kaya. Wala akong green screen.
Okay.
Pero yung...
And then, do you have a video in your presentation?
No, no.
Okay. Thank you, Miss Andrea.
20 minutes, diba? 20
Yes. Ay, dito si Ma'am Jacinto. Ma'am Jacinto.
Hi, Andrea.
Hi, ma.
Hello. Thank you, ha. Thank you for saying yes to our invite.
Opo.
Malapit ka na bang matapos?
Hindi ko pa po alam. Sana next year po.
Oh, pero at least meron na kayong mga publications, ha? Proud na proud si Ray sa'yo, eh.
Ilan po mamang participants today?
More than 100, I think. Kasi
Okay.
sa the previous 3 nights, maabot kami ng mga 126, ganon.
Tapos po yung Q&A after po ng talk. Tama po.
Yeah, there are 2 people and then there will be a Q&A after the 2. So ikaw and then there's somebody else and then may Q&A moderator and then maraming tanong. So kasi exciting yung mga talk. So lalo na ito kasi it borders on human responses, di ba? Or at least yung isang isang talk yata. So
okay.
how is it there? Parang dressed for really, really cold weather ka na dyan, ah.
Nasa lab pa po ako, ma'am, pero malamig na po ngayon.
Ang haba na ng hair mo! since
Opo.
the last time I saw you, parang napaka-short noon.
Opo.
Ang tagal na kita hindi nakita pala.
Matagal na po yung college.
Oo nga, oo.
Bio 11.
At saka di ba, nag-partner tayo nung project. Sino ba yung may project na, yung sa Amor, you were part of Amor, right? Amor 2.
Hindi po ako, ma'am, pero yung iba ko pong batchmates part ng Amor, yung mga cone snails, si Ma'am Santos at si Ma'am Monne.
Yeah, okay. Pero sila Miren at ano wala na sa new MBB eh. Parang naalala kong meron kaming partner na si Jay Lazaro ang head.
Ah, hindi po ako part ng lab
Hindi,
niya,
hindi.
ma'am.
Okay. Alright. O sige, ano ka na, mag-relax ka na dyan. And malapit-lapit na rin eh.
Sige po, ma'am.
Okay,
Balik po ako
sige
mamaya.
Andoy. Okay, sige.
Sige po, ma'am.
Bye-bye.
Bye-bye.
Hello po, mag-share screen lang po ako ng PowerPoint ulit.
Nawala si Ma'am Andrea?
Balik daw siya.
Okay. Okay.
Okay na po yun sa PSDB channel. Okay na po yun sa YouTube. Babalik ko na po kasi kay Ma'am Joyce yung hosting juries.
Yes po, ma'am.
Okay na. Salamat. Balik
Ano
ko
po
na
yung
sa
nakita?
Ay, sorry.
Sige po, Ma'am Red.
Ma'am Joyce, pagawa na lang po kung co-host. Thank you.
Okay.
Ano po yung nakikita niyo sa sa screen? PowerPoint po ba?
Co-host ka na, Lair.
Thank you, ma'am. Yung PowerPoint na file, nakikita namin, hindi presentation.
Okay po. Inverted po ba yung background ko? Hindi naman po.
Ay, wait.
Okay
Hindi.
lang.
Okay lang po.
Hindi. Bakit kulot din yung hair mo?
Ulat talaga ako, ma'am.
Nakikibagay kay Larry. Pareho din kayong kulay ng damit.
Oo nga eh.
Oh,
Ma'am Larry, ay naku,
Ano
ma'am.
Ano kayo?
Ano ba yan, ma'am?
Ano kayo?
Nag-usap kami, ma'am, kasi Q&A
Galing.
facilitator ako ni JV, ma'am.
Mhm.
Ayun na si Sir Ahmad. Hindi, co-host na si Sir Ahmad.
Teka lang. So, Amadeo, sasan ka?
Kinukulit ko na po kayo.
Amadeus, nakuha mo na ba yung ano po?
Ah,
Yung...
Saan nyo po kasi parang may tab dito na dapat pupunan niyo po. Did you send it sa ibang po? Sa ibang platform ba?
Teka lang. Hanapin ko.
Ayun, sa baba po kasi merong criteria tab, Jacinto scoring sheet, Ferrer scoring sheet, and the Chavez scoring sheet. Tapos may final tally tab. Ako na lang
Yeah. Kasi natapos ko na siya kagabi eh. So,
alright.
hindi ba yun automatic share?
Parang hindi yata.
Sige, teka ha. Teka, Amad.
Sige po, ma'am. I'm on it.
Sige, wait.
Hello, Ma'am Larry.
Yes, JV, I saw it.
Ano po ma'am, meron po palang prepared na video presentation si Ma'am Manilay.
Oo.
Yun po. So, pero attend naman po siya no, sa Q&A part na lang din po. Katulad po
Yes.
ni... Okay po. Thank you po.
Nandun naman nakalagay naman dun sa ano, na recorded presentation si Dr. Manila.
Yes po. Ma'am Joyce,
Yes po.
pwede po kayo yung mag-play ng video kasi yung laptop ko po mabagal. So baka...
Sige, try ko po mamaya kasi
Okay, sige po.
baka mahina din. Sige, sige,
Ah,
sige lang. Sige,
sige
chat
po.
na lang tayo.
Buksan ko na rin po yung video. Tapos if everyone po may mangyaring hindi inaasahan, may baka po.
Huwag naman sana. Sige,
Sige, ano ko din, iti-download ko din, JV.
Okay.
Nasa
Thank
ba
you
yung
po.
PowerPoint mo din, JV? Nandito din.
Ay, sinend ko po sa GC po ng session 4.
Yung PowerPoint mo?
Opo. Ay, hindi ko pa pala na-session.
Jeez!
Wala ko rin
Sponsor!
yun nga po. Hindi ko pa siya na-session pero nakalagay na siya dun sa...
Okay. Iba kasing account ang ginamit ni ma'am. Okay.
Oh, Ahmad, kasi send ko lang. Natanggap ko na.
I think so. Yes.
I shared it with Ryan de Chavez and Joyce. Si Joyce Ferrer, di ba?
A copy of it. Okay.
Is-
Yeah.
Sa Joyce Ferrer is the other one, tama?
Yes.
Okay.
Medyo nalagay lang kasi siya sa ibang ano. Sige, ako na bahala.
Okay, sige. Thank you, Ahmad.
Nalagay lang po sa ibang ano. Ako na bahala mag-ano nito. I assume po sa inyo to ma'am itong mga scoring na ngayon lang. Okay. Yeah, I see it.
Oo.
Nandito
Oo.
ka S, may S eh. Sige, ako na
Pero
bahala.
mamaya na yung 25%, di ba?
Yes po, after the Q&A.
Yes. Okay.
Okay po, I'll process this.
All right.
Wala pa, JV.
Yes po, ma'am. Wait lang.
Ay,
Sorry mo, ang bagal talaga.
Ay, by the way, lahat naman tayo natural products, no? Are you nabobombard ako ng mga emails from India eh.
Wala naman po. Sa akin.
Wala. Kasi parang I do not know, nag-call kasi ang DOST ng medyo gusto kasi nila na makipag sa grant. DOST yata yung naglabas ng call.
Oh. Bakit yung India?
Ito po kasi, hindi ba ma'am, minsan kasi merong mga call ang DOST na parang with an ASEAN country or parang with Asian ganun. May mga call silang nire-release na ganun eh. Parang it's supposed to be diplomatic
Oh. Kasi alam na siguro nila ikaw yung nag-head sa MCCL.
Yeah, pero I've heard kahit sa CHEM din eh, may mga ini-email din sila sa CHEM eh.
Naku.
Wala lang, curious lang ako. Pero hindi ko pa naman tinetake seriously at this point. At this point din, kasi syempre mahirap pag hindi natin kilala eh.
Correct.
Matrabaho din kasi gumawa ng proposal and then kung hindi naman sipag mo yung
Nakakatakot yan. Nakakatakot mga, sorry ha, sa India.
Opo.
Kaya, worry tayo.
May isa tayo ma'am, di ba, na dapat parang visiting professor din sana.
Si Pala Bhattacharya, hindi na siya nag-pan out eh. So si Mike, ang kausapin mo na, nag-usap kami ni Mike ng matagal. Tungkol dyan, kasi gumawa rin si Mike ng program eh. What he can be doing while he is here. Pero eventually, hindi naman natuloy. Okay, sige.
So, nasa tali lang ako. Ayos lang natin.
Ma'am Larry, wait lang po. Oh.
Konti pa lang yung nasa waiting room. Pero 9 o'clock pa rin. 9 o'clock pa naman. Pero
Okay lang yun. Ano naman yan eh. Ang ugali naman natin kasi talagang right on the button tayo minsan mag-login eh.
tsaka, tsaka
Oh.
Saturday.
At saka Saturday, yes. You have to put that into consideration. Gusto ng mga tao tanghali magising ng konti pag Sabado eh. Mm. Pero if you notice kasi, syempre, ang trend ngayon, pag online, mas marami ka talagang participants. Mas mahirap pag ano. Okay.
Hello po. Rinig po ba yung electric fan namin? Hindi naman po.
Hindi naman.
Okay. Ma'am Larry, send ko na po sa inyo yung PowerPoint sa chat box po ng Zoom.
Thank you.
At saka sa inyo din po, Ma'am Joyce. Sending pa lang po.
Okay, thank you. JV, parang wala pa doon sa chat box natin, sa group chat natin.
Hello, ma'am. Dito po sa Zoom na chat box. Naka-direct message po sa inyo.
Akala ko sa GC natin. Okay, okay. Dr. Manilay is here na po. Hello po.
Hello.
Hello, Dr. Manilay.
Hello.
Hi, it's nice to see you.
Nice to see you too. Can you hear me?
Yes, we can hear you okay.
Great. Thank you.
Thank you. So we will be starting in a few minutes. So we will just play your video. Do you want to introduce your video?
No,
Or
it's okay.
just go ahead?
I have it's already got an introduction. Thank you.
Okay, thank you.
Thank you.
Sir Judel.
Hello ma'am, good
Hi,
morning.
good morning. Sir Judel, okay ka na for yung photo contest ba? We will have a separate, 'di ba? We will have a separate for session, winner for session 4 and session 5.
Yes po ma'am.
Kahit yung winner na lang.
Okay po ma'am.
Ah.
Good po ma'am.
Tapos actually, hindi pa nga pala natin nilalagay with the session moderator. JV, yung photo contest, saan mo nilagay?
Dito po, ma'am, sa part na to. After ko pong
Okay.
after po ng conference schedule. After po no session 4, ma'am. Tapos ako po sasabihin tong part na to.
Pwede yung
Or-
winner.
Yung winner, ma'am, sa last part po, ay sorry, hindi ko pala nailagay. Ayusin ko
Ayun.
lang po yung after po nung poster live Q&A judging.
Okay. So yung pinakahuli before we go for lunch break, right?
Hindi naman po nakahuli, ma'am. Ay, huli na po, ma'am, siya. Kasi thank you for participation na po yung kasunod.
Oo. So, dyan na lang. Before
Wait
mo
lang po, ayusin ko lang po.
Okay. Thank you.
Diyos ko, nanginginig ako sa bagal ng Diyos ko.
So,
Ay, sorry, naririnig po pala.
oo. So, mag-play na ba tayo? We'll let them in na ba? And let's start playing the sponsors videos or in a few minutes. Kasi we'll announce na in the waiting room.
Ako po ma'am, 3 minutes po muna siguro.
Opo, ma'am.
Okay. So, mga 5 minutes? Is
Yes.
that okay? Okay.
Ma'am, kayo po ba yung mag-a-announce ng winner dun sa photo contest?
Pwede nandiyan.
Kanino ko po ito turn over, ma'am?
Pwede naman ikaw na mag-announce. Sabihin sa namin ni Sir Judel, yung winner na lang yung ipapakita.
I-share screen yun na lang po.
I-share. Papadalan ka na lang ng sige, ako na lang mag-share ng screen.
Okay po.
Sige.
So, ilalagay ko po siya, ma'am, ay dun po sa last part po after ng Q&A ng poster.
Yes. After nung Q&A ng poster, that's right. Right before you say thank you for the participation and
Okay, sige
we
po.
will have our lunch break, right?
Okay po.
Thank you.
So, photo contest po yun, ma'am.
Sir Judel, pasend na lang po sa akin yung pong winner. Ako na lang po mag-share. Thank you.
Pa-record po.
Teka, sa cloud ko ba ire-record? Or
Yes,
sa
ma'am.
Sa
Cloud,
cloud.
ma'am.
Okay.
Wait lang po.
Hindi pa naman magpa-play hindi pa magsa-start si JV. We can record later. Nag-aayos pa ata si JV ng PowerPoint niya.
Naririnig niyo po yung audio ng PowerPoint?
I think right now we can't, pero baka you have to share the sound. Hindi ka ata naka-share sound.
Okay. So, wait lang po ulit.
So when you have videos on your PowerPoint, pag nag-share
Okay
screen
po.
ka, pa-share sound. Thanks.
Share sound. Okay. Okay na po. Hello po. Naririnig niyo po?
Yes po.
Okay po. Inom lang po muna akong tubig.
Kailan ako mag-start ng recording, Lair?
Bali, ang mangyari po, we admit all, disable the waiting room.
Yes.
And then magpa-play, pwede na po nating i-record.
Okay,
While
sige
nagpa-play
po.
po yung videos.
Okay, sige.
Thank you.
Happy.
Hello?
Yes, Lair.
Pinost mo ba yung ano mo, JV? Or talagang parang paputol-putol siya? Is it only... Sige, tetest mo muna lahat, JV.
Walang sound, JV.
JV, can you hear us?
Yes, ma'am.
Are you going to play to, okay. Trial playing pa rin to, right?
Ano po, ma'am?
Nagtatry ka pa rin mag-play, so hindi pa tayo mag-admit. Kasi when you're ready, we will admit na.
Okay. Pero narinig niyo po yung audio, ma'am?
Walang audio yan.
Walang naririnig.
So play ko na. Sorry, ma'am. Wala po rin siyang audio sa akin nung dinownload ko.
Meron siyang audio kagabi. Ito na video.
Ma'am Joyce, pwedeng kayo po mag-play ng mga advertisements?
Test.
Pero may ano na siya. Parang may audio na ito ngayon.
Okay.
Teka.
PrimoStar 3, your robust yet compact microscope for digital teaching and routine lab work. 10 highlights in 90 seconds. Inspire your students in a digital classroom. With Zeiss PrimoStar 3, you can connect their microscopes to each other in a collective learning network. Zeiss PrimoStar 3 comes in predefined packages, so you have a choice of ready-to-go combinations. Activate Eco Mode and your microscope will go into a power-saving sleep mode if you haven't touched it for 30 minutes. Activate the Light Manager when changing lenses and the microscope will
JV,
Zeiss
nagahang
PrimoStar
sa'yo,
3, your robust yet compact microscope for
Nagahang ba
Yes,
sa'yo?
ma'am. Yes po. Yes po.
Okay. Huwag mo nang ilagay sa sa PowerPoint. I will play it. Pero I
Okay po.
I have the med test. I have the first one, yung with all the sponsors. Pero I don't have the MaCare one.
Send ko sa link, Claire.
Okay, thank you, ma'am.
Then, ma'am.
Yes.
Hello, ma'am.
Nakita ko na.
Hello, ma'am. Mag-start po ka after niyo pong i-play lahat ng advertisements. Then, saka po ako mag-share screen.
That's right.
Okay po.
Zeiss PrimoStar 3, your robust yet compact.
So I will just share ha. Paki-check na lang if you can hear it.
Zeiss PrimoStar 3, your robust yet
Yes
compact
po.
microscope for
Narinig
digital
po.
teaching and routine lab work. 10 highlights. Seconds. Inspire your students students in a digital classroom. With Zeiss PrimoStar 3, you can connect their microscopes to each other in a collective learning network.
Okay, admit na tayo. Let's start.
Okay, disable ko na po yung waiting room.
Admit all first and then
God bless us all.
Zeiss PrimoStar 3, your robust yet compact microscope for digital teaching and routine lab work. 10 highlights in 90 seconds. Inspire your students in a digital classroom. With Zeiss PrimoStar 3, you can connect their microscopes to each other in a collective learning network. Zeiss PrimoStar 3 comes in predefined packages, so you have a choice of ready-to-go combinations. Activate Eco Mode and your microscope will go into a power-saving sleep mode if you haven't touched it for 30 minutes. Activate the Light Manager when changing lenses and the microscope will remember the precise amount of light you've set for each lens position. PrimoStar 3 houses an array of clever features. The carrying handle lets you move it around safely, and all cables are neatly stowed away at the microscope itself. Fixed Kohler versions of PrimoStar 3 come pre-adjusted with a field of view of 20 millimeters and a 4x nosepiece. With full Kohler versions, you profit from a field of view of 22 millimeters and a 5x nosepiece. PrimoStar 3 is made of solid materials, so you can rely on a mechanically stable and resilient microscope. That built-in durability is reflected in our offering you the benefit of an extended warranty of up to 5 years. Tailor your microscope to your tasks and let your applications determine which accessories you need. For further information, visit our website. Zeiss PrimoStar 3.
So good morning, everyone. Welcome to the 13th Annual Scientific Conference of the Philippine Society for Developmental Biology with the theme of Diversity in Developmental Biology. So we completed 3 sessions. Discussing about life history, life cycle, Rafflesia anatomy and development, and evolutionary developmental biology. And for today's session, we will be having developmental immunology. My name is John Vincent G. Navalan, or JV Navalan, and I am your moderator for today's session. But before anything else, we would like to inform you that we are having a photo contest for this session. All you have to do is take a photo of yourself while watching the PSDB 13th Annual Scientific Conference, post it on Twitter or Facebook using the hashtags #PSDB13 and PSDB13ASC, and tag us at PhilSocDevBio. The most creative, funny, and interesting photos will receive a gift from PSDB. So for today, we'll be having 2 speakers from University of California, Merced and University of Virginia. And together with me is Dr. Larry Ann Ipulan Collet, who will be the facilitator of our Q&A portion. And before we start, I would like to reiterate the session rules. First, please turn off video and audio upon login. Type your questions in the Slido app, and the link for the Slido will be provided in the chat box. The Q&A facilitator will collate and read the questions to the speaker. Please write your name, affiliation, and the name of the speaker to whom you are addressing your question. And for PSDB members, links to the evaluation form will be posted on the chat box. And please submit the form at the end of each session. For those who are asking how to get a certificate of participation, first, you must be an official member of PSDB registered in the annual scientific conference, must submit at least 1 evaluation form in the evening session and the Saturday sessions. And lastly, take note that the evaluation forms will open at the end of the session until 11:59 p.m. And to give us a message, may we call on the PSDB advisor, Dr. Sonia D. Jacinto.
Thank you, JV.
You're welcome, ma'am.
Good morning, everyone. Good morning, Dr. Manilay and Dr. Tubi Andrea Salvador, esteemed officers and board members of the Philippine Society for Developmental Biology, and all the members and all participants and sponsors of this activity. As I indicated in previous conventions before, the role of developmental biology is far-reaching and touches all fields of human endeavors, including physiology, health and medicine, agriculture, natural product studies, ecology, taxonomy, and systematics. Hence, there is something for every biologist in this field, a kind of scientific hub in biology, and the Philippine Society for Developmental Biology, or PSDB, advances this science through its many activities over the years. With pride, I commend the present officers and members of the board of directors of the PSDB for the decision to hold another virtual annual conference for this year. The Society fearlessly carried out a very successful virtual convention last year when others feared to tread, what with the newness of the lockdown situation and the difficulties in the planning and implementation. So here we are again under the same circumstance. Indeed, the theme for today's convention, Diversity in Developmental Biology, is very inclusive. Hence, the oral and poster presentations cover a diversity of topics, from developmental studies of the very interesting parasitic plant Rafflesia to corals, duck genitalia studies, and developmental biology of the starfish, crown of thorns, and a touch on the aspects of development of the immune system for this session. One is sure to find something to perk up one's interest. Of course, an encounter with the scientists involved in these researchers would be a thoroughly exciting experience which should not be missed. PSDB webinars always offer opportunities to broaden one's horizons as well as strengthen and enrich scientific networks. We are happy to greet the many long-time committed members who are with us among the newly registered ones. The society is more than enriched by your presence. I am glad to be here as well to be part of this annual gathering of minds to further the interest in developmental biology in the country, notwithstanding difficult circumstances. Together we say, Padayon, push forward, and move more power to PSDB. Thank you.
Thank you very much, Dr. Jacinto, for that message. Now,
Welcome, JB.
Now, to give us a recap on what happened on the conference, may we call on the advisor or the president of the PSDB, Dr. Larry Ann Ipulan-Colet. Hello, ma'am. Good morning.
Hi, good morning. Good morning, JV. Good morning to our advisor, Dr. Sonia Jacinto, and our speakers, Dr. Manilay and almost Dr. Salvador, Miss Andrea Salvador. So as mentioned, our conference had 3 weekday sessions. We have concluded 3 weekday sessions starting last Wednesday, November 17, Thursday, and Friday, representing each session. So I have here just a short recap of what happened during the session 1. So session 1 theme is on life history or life cycle. We had Dr. Simon Caballes discuss the effect of the maternal feeding diet of crown of thorns starfish, a coral-eating starfish, on the subsequent feeding and growth of their larvae. And our second speaker, Dr. Ruel Bennett, presented on the diversity of Peronosporomyces from phylum Oomycota, and he discussed their thalli structure, evolution, life cycle, and habitat. And from this session, we learned that life cycle and diversity of these different species. And then session 2 is all about Rafflesia. We had 2 speakers who talked on the Rafflesia. So the first speaker was Dr. Bascos. She discussed the infection zone of the Rafflesia consoluae, which is primarily within the roots of the host plant. And she mentioned that the reproductive structures of this Rafflesia species can develop even in the absence of extensive infection. While we had an invited guest, Mr. Wicaksono. He's an Indonesian, and their official or their national flower is the Rafflesia. And he presented his efforts in vitro and in vivo cultivation of different Rafflesia species. Definitely after this session, we gained a lot of understanding about this very special parasitic plant. And session 3, which was yesterday, we had 2 speakers, Dr. Patty Brennan and Dr. Dara Orbach. So they discussed the effect of mating behavior on the development of the external genitalia. Specifically, Dr. Brennan discussed the coevolution of birds' external genitalia, while Dr. Orbach discussed the vaginal morphology of different marine mammal species. So we definitely learned that the mating behavior of birds and mammals co-evolve between their mating behavior defines the morphology of the external genitalia, and that they co-evolve between male and female. So now I think we will learn, so far those 3 sessions has been very successful and we learned a lot. And I'm very confident that the session 4 and session 5 will be again a very interesting learning experience for all. So good. Thank you very much. And please stay until the end. Happy learning.
Thank you very much, Ma'am Larry. So I know that all of you are very excited to see the presentation of our first speaker. So I would like to introduce our first speaker. Our first speaker obtained her PhD in Harvard University, focusing on transplantation biology and lymphocyte development, and finished her postdoc in developmental immunology at the University of California, Berkeley. And she has over 20 years of combined training, study, and research discoveries in lymphoid cell development. Within the past 15 years, she contributed extensively to the shaping of UC Merced's academic and research programs in biology, which are themed on quantitative and systems approaches. She made contributions to science through her publications and scientific studies. Currently, she is a professor and department chair of molecular and cell biology at the University of California, Merced, and trained PhD students and undergraduate students in diverse projects related to thymic development, B-cell maturation, hematopoietic stem cells, and immune development across the lifespan. Ladies and gentlemen, our first speaker, Dr. Jennifer O. Manilay. So she has a video presentation. I'm now turning over this to Ma'am Joyce. Ma'am Joyce. Hello, ma'am. Wala pong audio kaming naririnig.
For a while. Sorry for the technical problem.
Okay po.
For a while lang po.
Okay po. So, again, we would like to thank our sponsors for today's conference, Macare Medicals Incorporated, Merck, Phil Anaserv, and MedTest. Thank you very much. And again, we have a, okay na po ba ma'am? We have a photo contest, so pwede niyo siyang i-post sa Facebook and then Twitter using the hashtag PSDB13. Okay, so here's the presentation of Dr. Manilay. Thank you.
My name is Jennifer Manilay, and I am a professor at the University of California, Merced, in the School of Natural Sciences. I am very excited to be with you all virtually, and I thank the organizers for including me in today's program. Before I discuss my research, here is just a little background about me. I am a Filipina-American, and I am the first American-born person in my entire family. My parents are from Quezon City and Manila, and my husband's family is in Santo Tomas, Batangas. I was introduced to Dr. Larry Ann Ipulan Colette and the Philippine Society of Developmental Biology by Professor Richard Beringer, a Filipino-American geneticist at the University of Texas. I am very happy to be in the presence of other Filipino scientists, as we are still rare here in the United States. In today's seminar, I will begin with a short introduction to stem cells, then explain my general research interests, and then proceed to tell you about two related investigations from my lab that have revealed how changes in bone health can impact immune cell development in the postnatal bone marrow niche. I am a developmental biologist, and my current research projects require connecting elements of the fields of immunology, hematology, developmental biology, and stem cell biology. As developmental biologists, we define a stem cell by its ability to self-renew or produce another copy of itself, and its ability to differentiate or produce a terminally defined lineage. In my research, I have focused on the pluripotent hematopoietic stem cell in mice and the cell signaling pathways and cellular interactions that influence its differentiation into distinct hematopoietic lineages, especially B cells, which are the white blood cells that produce antibodies as part of the immune response. In mammals, hematopoietic stem cells reside primarily in the bone marrow and can be found in different bone types, including femurs, tibias, hips, ribs, sternum, and calvaria. Within the bone marrow, work from many groups in the last 15 years or so have revealed a dynamic microenvironment in which hematopoietic stem cells interact with non-hematopoietic bone marrow stromal cells that may support hematopoietic stem cell self-renewal and influence their differentiation into specific cell lineages. In today's talk, I will focus on the bone marrow stromal cells that have been shown to support B-cell lymphopoiesis, including mesenchymal stem cells, osteoblasts, and osteocytes. This is a cartoon that depicts the locations of different stromal cells relative to each other, as well as their locations relative to hematopoietic stem cells and B cells within the bone marrow cavity. Mesenchymal stem cells are found within the central marrow in close proximity to sinusoidal blood vessels in what is termed the perivascular niche. Mesenchymal stem cells are precursors of osteoblasts, which are bone-building cells and are located where the bone marrow and the bone meet in what is called the endothelial niche. Osteoblasts can differentiate into mineralized osteocytes, which are embedded within the hard part of the bone. Osteocytes are the cells that sense mechanical loading. In addition to their roles in bone development, mesenchymal stem cells, osteoblasts, and osteocytes can regulate hematopoiesis and immune cell development in the marrow via direct cell-to-cell interactions and via secreted growth factors. It is thought that immune cell migration to specific stromal cell populations within the bone marrow niche is important for normal hematopoiesis. All of this background information is relevant to my research questions. I am interested in basic research questions related to cell fate decisions and differentiation in the immune system. Recently, I have become more interested in changes that occur in the skeletal and immune systems during the course of normal aging. In my work, we utilize molecular and cellular approaches and in vivo transplantation models in mice, and rely heavily on flow cytometry and microscopy to answer our scientific questions. To give you some context on how I fell into this work, I will start with my first story on sclerostin. Osteoporosis is a disease that is often acquired with aging, resulting in weak and brittle bones that can lead to a risk of bone fracture. Osteoporosis is a problem worldwide and a major health issue for the aged. Most medications to control osteoporosis focus on inhibiting bone resorption by osteoclasts, but only recently new drugs that focus on bone building have become available. Recently, sclerostin antibody therapies have completed clinical trials and have been shown to be very successful in increasing bone density in osteoporosis patients, reducing future risks of fracture. Here is a micro CT of two bones, the first from phosphate buffered saline or PBS treated control mice, and the second from a mouse treated with sclerostin antibody. Examination of the scans shows significant bone formation within the bone marrow cavity, which is the site of hematopoiesis. Sclerostin, also known as SOST, is a Wnt signaling antagonist that is secreted mainly by osteocytes. Osteocytes are the deeply embedded mineralized bone cells that I mentioned earlier. Sclerostin binds to the Wnt coreceptor LRP4, 5, and 6, effectively blocking the activation of Wnt signaling. And the role of sclerostin in bone homeostasis has been an area of active research. Wnt signaling in bone is important for the development of osteoblasts from mesenchymal stem cells, and is also important for the differentiation of osteoblasts into mineralized osteocytes. It's been shown that loss of function mutations of the SOST gene or deletions of the SOST regulatory region result in excessive thickening of bone tissue in human diseases such as sclerostiosis and Van Buchen disease, which makes sclerostin a good drug target for osteoporosis. However, Wnt signaling is also known to be important for the regulation of hematopoietic stem cells and for B-cell development in the bone marrow. Just to review, Wnt signaling, when binds to its receptors LRP5, 6, and the frizzled receptors, results in activation of the beta-catenin complex to the nucleus, resulting in gene expression. In the hematopoietic niche, Wnt3A and Wnt5A have been implicated in preserving self-renewal of HSCs. Wnt10B has been shown to be a primary Wnt molecule that drives fate decisions in osteoblasts. Frizzled receptors are expressed on many cells and can bind to multiple Wnt proteins. And Wnt antagonists, such as DIPCOF1, secreted frizzle proteins, and sclerostin also regulate the activation of Wnt signaling in the hematopoietic niche. When sclerostin is present, it prevents the binding of Wnt to its receptors, resulting in inactivation of Wnt signaling. Given its known role in hematopoiesis and in B-cell development, we hypothesized that in mice that lack sclerostin, the mice would have alterations in hematopoiesis due to overactive Wnt signaling. This is a quick summary of our findings, which are all published and available with open access. Since 2012, we have published several studies examining the role of SOST deletion on immune cell development. Our first study identified increased apoptosis of B cells in the bone marrow of sclerostin knockout mice, but we did not detect any effects on Wnt signaling in the B cells. Instead, using transplantation models, we found that the B-cell defect in the global sclerostin knockout was non-cell autonomous, and we found that the levels of a B-cell growth factor, CXCL12, was decreased in the SOST knockout stromal cells. This led us to conclude that the bone marrow microenvironment was altered due to changes in the bone structure. We followed up this study with an analysis of conditional SOST knockout mice in which deletion was specifically targeted in MSCs, osteoblasts, and osteocytes. This analysis surprisingly revealed that sclerostin deletion at the mesenchymal stem cell stage and osteoblast stages resulted in B-cell developmental defects. What was also surprising was that deletion at the late osteocyte stage did not affect B cell development. These two studies focused on mice aged 12 to 16 weeks of age. In our most recent paper, just published earlier this year, we studied older SOS knockout mice, and this revealed additional hematopoietic defects in the knockout, including evidence of inflammation in the bone marrow of the mice. Taken together, our studies revealed sclerostin as a new mediator of crosstalk between the skeletal and immune systems. To our knowledge, our basic research studies are the only reports of the effects of sclerostin depletion on immune cell development and suggest that osteoporosis patients may exhibit signs of B cell defects or inflammation in the bone marrow, weakening their immune response. We are currently seeking new funding to investigate this further in humans. Next, I would like to present my second story on immune cell development in another mouse model, the von Hippel-Lindau conditional knockout mice. This work is currently under peer review in Frontiers of Immunology, but a preprint is available on bioRxiv. This project is driven by my current PhD student, Betsabel Chicana, as part of her dissertation work, and is also a collaboration with Professor Joel Spencer in the Bioengineering Department at UC Merced. We became interested in the VHL gene because deletion of the VHL in DMP1 expressing cells, assumed to be mostly osteocytes, resulted in a drastic dysregulation of bone homeostasis, resulting in an occluded bone marrow cavity filled with skeletal cells. The bone phenotype appears as early as 6 weeks of age, earlier than the phenotype of the sclerostin knockout mice. Similar to the sclerostin knockout, the VHL knockout mice displayed a severe reduction in B cell development in the bone marrow. However, in the VHL knockout mice, this extended to the peripheral blood and spleens. We wish to identify exactly where B cell development was slowed. To do this, we utilized flow cytometry to identify the developmental stages of B cell development described by Richard Hardy in his seminal 1991 paper. This is a chart showing the different fractions, which are Hardy identified as fraction A through fraction F. And these are the cell surface markers that can be utilized by flow cytometry to identify these different stages in the bone marrow. We used this strategy to analyze B cell development in the VHL knockouts. To summarize quickly, this analysis of VHL knockout mice identified a severe reduction in B cells beginning at the earliest stage, fraction A. This was further verified in reciprocal bone marrow chimera experiments, in which wild-type bone marrow was transplanted into lethally irradiated control or knockout mice. Donor-derived B cell development was tracked by the expression of CD45.1, and this showed that B cell development was significantly reduced at fraction A and all stages downstream. Importantly, these experiments confirmed that the defect was non-cell autonomous to the B cells, as VHL knockout bone marrow cells transplanted into wild-type mice developed normally. To investigate the mechanisms underlying the altered B cell development, we again used flow cytometry to quantify apoptosis and proliferation of the B cells by their developmental stage. We found that fractions B and C were significantly higher in frequency at the late stages of apoptosis, and there were fewer cells at this stage within the SG2M cell cycle stages in the VHL knockout mice compared to controls. To investigate the changes in the bone marrow microenvironment that contribute to the B-cell developmental defect, we used a bead-based cytokine array to quantify several cytokines in the bone marrow fluid. We found that the B-cell growth factor CXCL12 to be reduced in the VHL knockout, but stem cell factor, another B-cell growth factor, was unaffected. These data are consistent with the reduction of B cells starting at fractions A through C. VHL deletion is known to stabilize hypoxia-inducible proteins, or HIFs. We hypothesized that the bone marrow microenvironment of the VHL knockout mice to be hypoxic, and this could contribute to the B-cell defect. Consistent with this, elevated levels of erythropoietin, or EPO, in the VHL knockout mice supported our hypothesis, as well as an elevated number of red blood cells, which is not shown here. To investigate oxygenation of the bone marrow microenvironment, we utilized the hypoxia cell marker pimodinazole, or PIM. PIM staining in combination with specific cell markers can identify whether a cell is experiencing hypoxia. To verify that our staining worked, we first examined PIM staining on LSK cells, which comprise hematopoietic stem cells and progenitor cells, and have been previously shown to reside in a more hypoxic environment compared to other hematopoietic cell types. The PIM staining in control mice is shown in blue, and the PIM staining in knockout mice is shown in red. LSKs in control and VHL knockout mice stained positively for PIM at both 10 weeks and 6 months of age. However, at 10 weeks, LSKs in the VHL knockout stained less brightly with PIM than controls, whereas at 6 months, the VHL knockout LSKs stained more brightly than controls. This indicates that LSKs experience a more hyperoxic environment at 10 weeks and a more hypoxic environment at 6 months. We next examined PIM staining on cells that express B220, which is a marker expressed on all B-cell Hardy fractions. Similar to the pattern on LSKs, B220 positive cells at 10 weeks stained less brightly with PIM, and B220 positive cells at 6 months of age stained more brightly with PIM compared to controls. Taken together, these data indicate that a change in the bone marrow microenvironments of the VHL knockout going from hyperoxic to hypoxic with age. We then disaggregated the B220 positive B cells into the individual Hardy fractions. We found that fraction A cells stained brightly with PIM compared to the other fractions. In general, this indicates that fraction A cells reside in a more hypoxic niche, similar to LSKs. However, in VHL knockout mice at 10 weeks of age, the PIM staining was not significantly different compared to control mice. In contrast, at 6 months, PIM staining in fraction A cells was significantly brighter in the VHL bone marrow microenvironment. This indicates that fraction A cells in VHL knockout mice experience a hypoxic bone marrow microenvironment, and this could explain their B-cell defects. We are now following up on the mechanisms underlying the changes in oxygenation. In collaboration with Dr. Joel Spencer's bioengineering lab, we are utilizing in vivo and ex vivo 2-photon laser scanning microscopy to identify the location of fraction A and B cells within the marrow of VHL knockout mice. This is just a simple example of our studies, which shows B cells labeled with green fluorescence protein, or GFP, and that they are localized mainly in the metaphysis of control animals. But in the knockout, the B cells are located closer to the diaphysis. We hypothesize that changes in local oxygen tension result in hypoxia in the niches of the VHL knockout bone marrow and contribute to the severity of the defect in their B cell development. Work to test this hypothesis is currently in progress. Using a UDisco clearing protocol, we succeeded to make an optically clear, intact long bone and captured all femoral vessel types, including arteries, sinusoids, and transition zone vessels at different depths. Quantitative analysis of the cleared femurs suggested that regardless of whether they were in close proximity or distant from the endosteal surface, blood vessel diameters in the VHL conditional knockout are significantly larger than the control group. We further hypothesized that changes in vascular permeability could contribute to the B cell defects we observed in VHL conditional knockout mice. To test this, we injected mice retroorbitally with 70 kilodalton rhodamine B dextran. Vascular leakage can be observed by the presence of fluorescent rhodamine B dextran in the marrow spaces surrounding blood vessels. We show representative maximum intensity projections of intravital Z-stack images in control mice and in VHL knockout mice within the calvaria bone marrow. We quantified vascular leakage by comparing the fluorescence intensity in the perivascular space to the fluorescence intensity in the adjacent vascular lumen 10 minutes after injection. We found increased vascular leakage in the VHL knockout mice. These results reveal that vessel blood barrier is altered and suggest that B cells in the VHL knockout may be exposed to more plasma and protein factors.
Mas big. Buti na lang dili ako ang kuan ron. Ito kagadi medyo ako.
In conclusion, our data suggests that reduced B cell development in the VHL knockout mice results from a reduction in CXCL12, an increase in erythropoietin, and local hypoxia for fraction A cells. In data I have not shown you today, we have also observed an increase in vasculature, vessel diameter, and vessel leakage, which are consistent with hypoxic microenvironments. In future studies, we are investigating the changes in supporting niche cells. Specifically, we will examine mesenchymal stem cells, osteoblasts, and endothelial cells. And we are planning to directly measure the oxygen tension in the bone marrow. To summarize my seminar, I hope that I have convinced you that crosstalk between the skeletal and immune systems is important in maintenance of B lymphocytes in the bone marrow of adult mice. And I hope I have convinced you that skewing of bone homeostasis towards bone building can adversely affect B lymphopoiesis over time. Evidence for this comes from our analyses of sclerostin antibody-treated wild-type mice, global sclerostin knockout mice, and in mice in which hypoxia response pathways are constitutively active in osteoblasts and osteocytes. These findings have clinical implications on the immune status of patients receiving new anabolic therapies for osteoporosis. Currently, we are in the process of generating novel inducible tissue-specific VHL conditional knockout models. Our studies to date have utilized the DMP1 Cree conditional knockout mouse, which despite its widespread use in skeletal biology research, is known to have off-target expression in non-skeletal tissues, and is also now known to be expressed in MSCs and osteoblasts. In addition, the DMP1 Cree model is active from ontogeny and is not temporally inducible. To resolve these limitations, we are generating tamoxifen-inducible Cree mice controlled by the LEPR and Taglin promoters, which have been shown by single-cell RNA-seq to be expressed in mesenchymal stem cells and early osteoblasts. We will then cross these with our VHL fluxed mice and analyze skeletal and immune system development after tamoxifen induction in adult mice. Single-cell RNA sequencing of bone marrow cells and skeletal cell types has revealed more heterogeneity in the previously defined subsets of MSCs and osteoblasts that could support immune cell development. As we move forward with our studies, we are hoping to also incorporate single-cell RNA-seq as part of our analyses to identify transcriptional changes within cell clusters in order to explain the dynamic mechanisms underlying skeletal and immune crosstalk in the bone marrow niche. Lastly, I am seeking new funding to further investigate the impacts of sclerostin depletion on the immune system of aged mice. Mice ranging from 18 to 24 months of age correlate with humans ranging from 56 to 69 years of age, which is around the age sclerostin antibody therapies for osteoporosis are administered to human patients. I am proposing a comprehensive study of skeletal and immune changes in aged mice that receive sclerostin antibody treatment, and also the creation of temporally inducible sclerostin knockout reporter mice for flow cytometric and ex vivo imaging of bone marrow niches. I would like to thank all the people who have collaborated with my team on this work, especially Dr. Gabriela Lutz from the Lawrence Livermore National Laboratories, the Vivarium and Flow Cytometry Core facility staff at UC Merced, and our newest collaborator, Dr. Joel Spencer, and his team. The research in my lab has been performed by an awesome team of undergraduate and graduate students at UC Merced, and we have been funded by several local, state, and national funding agencies. Here's a recent photo of the Manilai and Spencer labs outside of our new building into which we will move in 2022. Thank you for attending my seminar, and I hope you will come visit us in Merced as soon as you can. Maraming salamat po.
Thank you very much, Dr. Manilay. So I think that some of our participants have their questions. And if you have any questions, you can type or you can post it on the Slido. The Slido link is provided in the chat box. And we are also informing everyone that we are live via YouTube. The link for the YouTube live stream is also provided in the chat box. And before we introduce our next speaker, we would like again to thank our sponsors, Merck, MedTest, McCare Medicals Incorporated, and Phil Anaserv. So for our next speaker is a PhD candidate in biomedical science, neuroscience of the University of Virginia, where she was a recipient of Peach Fellowship Award in 2016. She is currently a visiting student on immunobiology at the Washington University in St. Louis, and she is an alumna of the University of the Philippines, Diliman, both for her master's in molecular biology and Technology in 2015, and bachelor's degree in molecular biology and biotechnology in 2008, where she graduated as summa cum laude. Prior to pursuing her PhD, she taught undergraduate laboratory classes, including molecular biophysics, principles of gene manipulation, and animal and plant tissue culture at the NIMBB UP Diliman, and also worked as university associate in various laboratories in UP Diliman, particularly in the disease, molecular biology, and epigenetics laboratory, protein structure and immunology laboratory, and Marine National Products Laboratory of UP Diliman. She made contributions to science through her publications and scientific studies in the field of meningeal lymphatic vasculature and neuromodulation by the immune system, focusing on cytokines. Our second speaker, Ms. Andrea Francesca M. Salvador. Good morning, ma'am. Good morning, ma'am. Hello po.
Oh, sorry. Ayun.
Good morning.
Good morning. Hindi ako makashare ng screen. Ayun. Oh.
For a while po. Okay na po, Dr. Andrea.
Thank you. Oops. Kita po ba yung screen? Hello?
Yes, ma'am. Opo.
Wait lang po. So good morning, everyone, and actually good evening from St. Louis. I want to thank the organizers, especially Ma'am Sonia, for inviting me to give a webinar today. And I'm really excited to talk about the work that we've been doing in our lab, focusing on neuroimmunology and particularly neuromodulation by the immune system, which we will try to focus on cytokines. So as we know, an organism encounters many stresses every day and perturbations to its homeostasis. So in this, cartoon, we see that this mouse in the middle is encountering like multiple threats. There are viruses in its surroundings, pathogens, helminths. There's threat related to the environment, like changes in temperature or some changes in homeostasis when it feels hunger or thirst. There's also threats related to the presence of predators, as depicted here by the owl. And in order for an organism to survive, it has to counter these stresses and perturbations to its homeostasis. Oops. And this is not an exhaustive list, but among the adaptive responses of organisms to common major threats are the following. So for example, when an organism encounters predators, they can either fight or flight the predator. When it encounters injury, tissue repair mechanisms must be engaged to repair the injury. And when you have food scarcity, there's foraging behavior. Same with thirst, you have water-seeking behavior, and the animal, in response to different changes in temperature, need to warm up or cool down. And in response to infection, the immune response must be activated to counter the infection or the pathogen. So these responses are mediated by the following response systems. And as we can see, they're mostly related to the nervous system or the brain, and the peripheral nervous system and the immune system. So both of these systems, the immune system and the nervous system, mediate adaptive responses to threats to an organism's homeostasis. And these are among the characteristics which make these systems able to mediate these challenges. First of all, both systems have the ability to recognize perturbation in their environment. The immune system, for example, is composed of immune cells, and these immune cells express receptors on their surfaces that can detect pathogens, which are non-self or strangers, or they also have receptors that can sense damage. So these might not be related to the presence of an a pathogen, but like tissue damage or other like stresses in its environment. The nervous system also is able to recognize perturbations because they express different receptors and ion channels on its surface. So it's been reported that neurons express cytokine receptors, and they also express the same receptors as immune cells to detect pathogens, and they also express different ion channels that can detect noxious stimuli. So if you remember, like this year, the Nobel Prize winners were for the discovery of the TRP channel, which senses heat and capsaicin and mechanotransduction ion channels. So both of these systems can recognize perturbations. They both also display a high degree of plasticity. So in immunology, we know that T cells are able to differentiate into different effector cells. Macrophages are also extremely plastic. And the nervous system is also endowed with a high degree of plasticity, which we will discuss further later. And they're also pervasive and widely distributed in an organism, which makes communication of different parts efficient. So for example, the immune system, the immune cells circulate in the body, and the nervous system is the signal is transmitted by electrical signals, which is fast acting. Despite these common traits, the nervous system and the immune system, they have different strategies to counter threat. So the nervous system, as we know, controls the expression of behavior. So in this very simplistic diagram, we can see that, again, this mouse may experience some change intrinsic stimuli, which is hunger or thirst, or the presence of an extrinsic stimuli, which is a predator. And upon sensing the stimuli through their sensory organs and then processing and integration in the different brain regions, they can mount an appropriate behavioral response. So in response to hunger, they can seek out food, or in response to the predator, they can flee from the predator. And what's important is that the nervous system, when they receive the cues from the environment, they are able to process and integrate this information so that they can mount an appropriate behavioral response. So as we know, the neural circuits influence expression of behavior. So there have been many studies in the past which they perform loss of or gain of function studies using lesion studies, and through the development of optogenetics and chemogenetics, where you can specifically target circuits of the brain without causing massive injury. These have allowed us to determine the brain circuits that mediate certain behavior. So as an example, the limbic structures in the mouse brain, which is composed of the hypothalamus, amygdala, and the hippocampus, they mediate different motivational behavior, such as sexual behavior, defense against predators, and conspecifics, and maternal care. But these neural circuits, which are anatomically wired, aren't as flexible, because once you're born with like the wiring of your brain, it won't really change because neurons are post-mitotic, and when they die, you can't really replace them unless you're in the periphery or in some other parts of the brain. So how is the plasticity or the flexibility of the nervous system achieved? And neuromodulation allows the flexibility of neural circuits so that organisms can respond to different stimuli by adaptive behavior. So neuromodulators can either be neurotransmitters or neuropeptides that act locally or distally, and they can modify the properties of neural circuits to reflect an animal's global state. So for example, you're stressed, or when you have like hormones from your adrenaline, it can cause you to change your behavior, and that affects your neural circuit so that you can respond to it differently. Or when you are feeling some pain, you react in a different way. So in this diagram, you can see that every process of the neural processing, from sensing and the processing and integrating to your brain to the effector function, can be modulated by the different neurotransmitters or neuropeptides. So unlike the nervous system, the immune system has a different strategy in countering threats. So once I encounter a pathogen, for example, through the pattern recognition receptors on the surface of immune cells, these immune cells are engaged and they activate an immune response so that they can clear the pathogen efficiently. And as we know, there's an innate arm of the immune system, which takes place in a matter of hours, and there's also an adaptive arm of the immune system, which take up days to develop, and they can interact with each other so that an effective and a long-lasting immune response is achieved. And among the modules that are activated by the innate and the immune system, among these modules and among these factors are cytokines. So cytokines are small molecules, and they can act in a specific way depending on its binding to a specific receptor. And what we're trying to argue is that cytokines act as neuromodulators. So one of the most characterized example of this is during infection. So when we're sick, or when you got like your vaccine, you experience a set of symptoms called sickness behavior, which is characterized by reduced mobility, motivation, appetite, and this is usually accompanied by fever, which is depicted in this diagram here. And people have studied sickness behavior in animals, and by either infecting them with a bacterial or viral pathogen, or by injecting them with LPS, which is what's this? The proteoglycan outside like bacteria. So the immune system recognizes the pathogen or LPS or poly-IC, for example, and then it releases a slew of cytokines, more commonly pro-inflammatory cytokines. And aside from sickness behavior, it can also induce depressive behavior in mice. And this is shown here. So this is the forced swim test, where you put a mouse in a jar and a beaker where it can't escape, and then you measure its escape behavior. So when mice are more depressed, they stop moving and they stop trying to escape. So when you infect mice with the virus, VSV, the immobility time is increased compared to those that weren't infected, suggesting that these mice probably have some depressive behavior. Aside from depression and sickness behavior, the infection also causes some learning deficits measured in different behavioral tests in rodents. But you can also argue that these learning deficits are induced by the reduced mobility of the mice, so if they can't move, maybe they won't be able to learn. But what's most important to note is that the infection is linked to pro-inflammatory cytokines, particularly IL-1 beta, TNF alpha, IL-6, and interferons. People have studied this because when people inject IL-1 beta, for example, in wild-type mice or healthy mice, it's sufficient to cause sickness behavior symptoms. And if you remove some of the receptors for these cytokines, it can also attenuate the sickness behavior. So if cytokines are neuromodulators causing sickness behavior, how then does the brain sense the immune system or the inflammation? So there are different routes for the brain to sense inflammation. First is the neural route. So when you have a peripheral infection, the infection can either activate sensory neurons outside the brain, and then by binding to either cytokine receptors, by engaging these ion channels, or by binding, because these neurons express the pattern recognition receptors. Upon binding, these peripheral neurons are activated and they signal to the brain. And one of the routes that has been described is through the vagal route. And vagotomy, actually, when you inject mice with IL-1 beta, for example, and you cut the vagus nerve, there's a reduction in sickness behavior, suggesting that this route is important for relaying that information to the brain. So aside from the neural route, there's also a humoral route for the brain to sense inflammation. As I noted earlier, immune cells circulate in the body and they release cytokines. So the cytokines are present in your blood, and these cytokines can then enter the brain through its leaky barriers called the circumventricular organs, or they can engage the receptors on the barrier cells, such as epithelial cells and endothelial cells. And these cells in the barriers make other soluble factors like prostaglandins, and these affect neurons so that you have changes in behavior. It's also possible that these cytokines can directly engage neurons to cause the changes in behavior. And I'd also like to mention, this is what our lab is really focused on, is some cytokines are derived from the meninges, and from the meninges, it goes to the cerebrospinal fluid and it can engage neurons. So before I talk about other examples of neuromodulation, I'd like to talk about the meninges and the borders of the brain. So this is just a depiction of the anatomy of the brain. So you have your brain parenchyma, and it's encased by your skull. And outside your parenchyma are is your meninges. And the meninges is a triple layered membrane. The closest layer to the brain parenchyma is your pia mater, and then you have your arachnoid matter and your dura mater. So below the arachnoid matter is the subarachnoid space, which is where the CSF flows. And then you have skull, and the skull has also bone marrow. And it's interesting because the previous speaker talked about the bone marrow. So anyway, what's important to note here is that the dura mater and the borders of the brain is host to a diverse array of immune cells depicted here. So the healthy meninges, in the absence of any infection, contains T cells, B cells, NK cells, macrophages, innate lymphoid cells, dendritic cells, and neutrophils. And these cells can make cytokines that can diffuse to the CSF and affect neuronal function. This is how the cytokines are going from the meninges to the brain. It's still a matter of active investigation, but that's really something that we are interested in the lab. And in this image, you can see, so this is a whole mount of the meninges. When you peel it from the bone and you lay it flat in a slide, you can see that there are CD3 positive cells in the sinus. So here in the sinus of the meninges and outside the sinus, just showing that the meninges is an active immunological site. So unlike the brain parenchyma, its borders are populated by immune cells at steady state. So we're interested now in, is there cytokine neuromodulation beyond inflammation? So in the absence of a pathogen, can cytokines affect neuronal function? So this has been shown in mice deficient for cytokine receptor cytokines, or mice that don't have an adaptive immune system, so they don't have B cells or T cells. These mice have been shown to have some behavioral deficits, which suggests that there probably is some homeostatic function of cytokines. So I'm just going to go through some of the examples that have been described in the literature and have been shown by our lab. So T-cell-derived cytokines support learning behavior. So learning behavior is measured in mice using different tests. One of those tests is called the Morris water maze, where you put mice in a tub of opaque water, and they have to determine where the platform is. And over the course of the test, the time that it takes a mice to find the platform goes down, which means that they're learning. So if you deplete mice of T cells, either through the genetic mutation of PRKDC, which is important for the maturation of lymphocytes, or you deplete them by blocking an integrin, or you block their exit from the lymph node, we see that there's impairment in learning in mice. And you can also test learning using the Y maze. So ideally, mice that will learn will alternate between the different arms of the Y maze. But if you deplete this specific T cell, which is called the gamma delta T cell, or you deplete the cytokine that is produced by this T cell, there's a deficit in short-term memory, which is measured in the Y maze. And more recently, we published, like it was 2 days ago actually, when it came out, that mice, when you deplete them of T cells, can't learn contextual fear learning. So in this test, you put the mouse in a box and you give them an electric shock. And then when it learns, and if you put it in a box without the shock, but in the same context, it will freeze. So the mice that are depleted of CD4 T cells have less freezing, so they don't form the same memories compared to wild-type mice, or compared to mice that have their T cell repertoire intact. And we also observe this in mice that are deficient for IL-4, and in skid mice, which we've previously shown, but this is just a different task. And what's most interesting is in skid mice that are deficient for the learning behavior, if you give them T cells that are IL-4 sufficient, then they can actually rescue that learning behavior. So there are several proposed mechanisms as to how how cytokines support learning behavior, and some of these mechanisms suggest that IL-4 acts through glial cell or astrocyte, and these astrocytes make BDNF, which is a growth factor for neurons, and this promotes neurogenesis, which is similar to this mechanism where the cytokine acts on some other responder, and this responder acts on neurons. But what we've shown in our recent paper is that IL-4 receptor expression on neurons specifically is actually critical for learning. So when you delete the IL-4 receptor in synapsin positive cells, which is a marker for neurons, you also see a decrease in learning, suggesting that the expression of this receptor in neurons is important. And if you inject a virus in different brain areas that are important for learning behavior, such as the hippocampus and an area of the cortex, you see that there is a decrease in the freezing or deficiency in learning behavior. So in this paper, we show that in homeostasis, T cells, which make IL-4, will act on IL-4 receptor expressing neurons to maintain its ability to learn and some synaptic properties. So I'm not going to show you that, but it's probably show, if you want to ask me later, then we can talk about it more. So aside from learning behavior, we've also shown that other cytokines from T cells also support social behavior. So how do we test these in mice? So this is called the 3-chambered assay, and you put your test mouse in the middle, and in one chamber there's a novel object, and in one chamber there's a novel mouse. So mice are very social creatures, so ideally wild-type mice will associate more with a novel mouse compared to the novel object. And we've shown previously that mice, the SCID mice that I've mentioned, they have deficiencies in this social behavior. So here, so as I mentioned, wild-type mice will typically interact more with a mouse versus a novel object. But in mice that are deficient for T cells, there's no preference for the novel mouse. But if you repopulate the SCID mice with T cells, here in blue, you show that you rescue the preference of your test mouse with the novel mouse. And among the cytokines that we tested or we surveyed, we show that interferon gamma is actually important for maintaining this social behavior. And what's also really cool is that if you delete the interferon gamma receptor, specifically in neurons, in a specific area of the brain called the prefrontal cortex, it is sufficient to impair the preference of the mouse to the novel mouse. So suggesting that the interferon gamma acts on neurons to support this behavior. And the proposed mechanism for this is the following. So T cells make interferon gamma, and the interferon gamma acts on neurons in the prefrontal cortex, and this will increase in the inhibitory currents so that it will be maintained. But if you have a depletion of the interferon gamma receptor in neurons, you see that there's more excitability because there's less inhibition of these neurons. And in the light of evolution, we saw that aggregated animals or social animals such as mice, I think these are rats and flies and fish, they have an enrichment for the interferon gamma transcriptional signature, suggesting that maybe because when you are in social situations, you are more prone to getting infection. So maybe you have to develop this immune response, especially gamma, because it's a powerful cytokine, so that you can combat infections properly. So they probably co-evolved together to support the social behavior while still maintaining its organismal fitness. And we kind of proposed this theory where what is this? Drosophila are infected by a retrovirus, so that they will develop this STAT1 signature, which STAT1 is downstream of interferon gamma, and the STAT1 signaling will be vertically transmitted, and these will make the organisms more social because they are more they have increased fitness, and this allows like humans, mice, and fish, which are social, to be social, so that they are protected from the infections you have that you might have from social interactions. So lastly, I want to talk about another example of how cytokines influence behavior. And as I mentioned, gamma delta T cells, which are T cells that are usually located in barrier tissues, and they make different kinds of cytokines. They regulate anxiety behavior. So how do we test anxiety behavior? We use the elevated plasmase. So because mice are fearful of predators, they prefer to be in closed areas of the maze, as opposed to the open arms, because in the open arms they'll be more visible to predators. So in mice that have deficiencies in these T cells and in one of the cytokines they produce, which is IL-17, these mice have less anxiety behavior. So they seem to be more risky because they populate the open arms more compared to the closed arms. So this is shown here. So as I mentioned, wild type mice will typically spend more time in the closed arm of the maze as opposed to the open arms. But if you deplete them of this specific kind of T cell, there's really they seem to populate the open arms more compared to the closed arms. And if you actually give an antibody that will block the function of the gamma delta T cells in the CSF, so hopefully it will block the meningeal gamma delta T cells, it will also reduce their anxiety behavior. And this is the proposed mechanism for it. This is still something that we want to pursue, so we're not sure how it really works. But what's most interesting is that IL-17 signaling on neurons actually control anxiety-like behavior. So meningeal gamma delta T cells produce IL-17A at baseline. So see here in our flow cytometry plot and using a IL-17 reporter mouse that they produce IL-17A even without any infection at baseline. And in the mice that are deficient for the gamma delta T cells, you see a decrease or loss of the IL-17A production in the CSF. So it suggests that maybe if you give mice, if in these mice without gamma delta T cells, but if you give them IL-17A, which is something that they need, maybe it will rescue the decrease in anxiety-like behavior. So this seems the case because what is this? They seem to be populating the open areas less compared to the gamma delta deficient animals. And if you deplete, if you remove the receptor of IL-17 receptors in neurons, specifically in the prefrontal cortex, you also see an increase in anxiety-like behavior because they populate the open arms more. So IL-17A is an important immune cytokine and it's present in barrier tissues. And aside from controlling neuronal function, it has other functions in mammals and C. elegans. So it has been shown in mice that IL-17A supporting sympathetic nerves in the adipose tissue to prevent to promote thermogenesis and prevent obesity. It's important for learning and it's important for controlling anxiety-like behavior. And in C. elegans, it's been shown that a homologue of IL-17A, IL-17, is being produced and it signals to neurons so that worms can aggregate, can avoid, nauseous stimuli. So in summary, I just want you to appreciate, hopefully, that there are non-immune roles of cytokines in controlling an organism's behavior, thereby acting as neuromodulators. And cytokines may be derived from the brain's borders, especially the meninges. But there's still really important unanswered questions with regard to this work. So are there other behaviors, for example, that are influenced by cytokines? And are there neuronal circuits more susceptible to cytokine modulation? So the advancement of single cell technologies can really help us answer these questions, because there's a lot of data out there where you can look at different mouse brain neurons, for example, and look for cytokine receptors and then kind of predict, because some circuits are known to be important for this behavior, then you can link them together. And this is one of the analysis that we did, where we looked at the cytokine response signature of mice and some of the and their responses to different stimuli, and kind of predict what behaviors or what stimuli are being controlled by these cytokines. And it also suggests that there's probably some close evolution of both systems. So there's an evolutionary arms race of the presence of pathogens, for example. What is this? Put pressure on the immune system to evolve so that you have like a more specific and a more potent and more long-lasting immune response, which is mediated by the somatic hypermutation of your lymphocytes, so that it can detect more pathogens. And we think that the development of a more advanced immune system has allowed the evolution of a more complex brain, because we think that because the brain is very fragile and it's poorly regenerative, so you probably need support from the immune system for some like another protection, so that you have a more complex behaviors. And maybe complex behaviors force a evolution also of the immune system, as I mentioned, maybe because humans become more socially humans are engaged in social behaviors, they needed support from the immune system so that they won't be able to succumb to infections that you gain from these interactions. And lastly, so I was talking about how the immune system is affecting the brain. People are also interested in the neural regulation of the immune response. So neurons in the periphery, for example, can modulate the immune response. It can control the velocity of the immune response by calling more cells to where you have an infection, by innervating, for example, your blood vessels. It can also regulate the immune response. So there are different neuropeptides that can suppress or can potentiate the immune response, which are derived from neurons. And they're also important for the resolution of the immune response. So, like for example, when you have spinal cord injury, there's a global immunosuppression, which is probably a response of your body to that trauma, so that you won't have like pro-inflammatory cytokines in your injured area, and you can have like more wound healing. And another important questions that we're tackling is that, is there a central representation and regulation of the immune response? So we know that the immune response is controlled by neurons in the periphery, but is there a center in the brain, for example, that collects information from your immune system and somehow controls the immune response globally? So with that, I would like to acknowledge my lab, which most of the studies I've highlighted were from my group, and yeah, I would be happy to take questions. Thank you.
Thank you very much, Miss Andrea. That was a very interesting talk. So can you hear me?
Yep.
Okay. Okay, so it's very interesting to know that the immune system and the immune system nervous system has that kind of interaction with each other, especially controlling behavior. So it's my first time to know that. So first, can I ask for Dr. Manilai's video to be turned on? So can you unmute yourself also?
Can you hear me?
Yes, we can hear you. So we
Hello.
have several questions here for both of you. So I will start with questions for Dr. Dr. Manilay. Is there an inherent difference in Wnt signaling in the immune system of young and old mice?
So we haven't noticed any, we didn't test specifically Wnt signaling in young and old mice, but in the sclerostin knockout mice where we studied them at 12 to 16 weeks, we didn't see any changes in the Wnt signaling in the B cells and the hematopoietic cells. What we did notice was that there is a difference in Wnt signaling in the osteoblasts, so the bone cells. So we think that the changes in B cell development, the diminished B cell development that we observe in the sclerostin deficient mice is actually an indirect effect of changes in the osteo lineage cells. Oh, did we freeze?
Okay. I think my connection is currently unstable, but I heard you. It's okay. Thank you. Thank you for that answer. So let's have a question from Miss Andrea. So this is kind of a tangential question, but someone in the participant is interested to know whether you think the psychobiotics or probiotics with psychological effects, sorry, with psychological effects will be really significant neuromodulators in the future.
I mean, it's possible because I think more and more people are, appreciating how the gut and the brain are connected. I feel like when you change I think maybe probiotics change your microbiome, and maybe if you change your microbiome, you're affecting some basal inflammation in an organism, and maybe that's signaling to your brain. And I think aside from cytokines, people have also looked at metabolites as being neuromodulators. For example, what's this? In sickness behavior, it's been shown that kynurenine or IDO, they also promote, which are both metabolites, are both causing sickness behavior. So maybe that's how probiotics can change behaviors eventually. But I think it's really exciting. And there's also an enteric nervous system that people are studying right now, and maybe that's connecting the body and the brain.
Okay, thank you for that answer. So for again, Dr. Manilay, what cells express the VHL and how is this normally regulated?
Sorry, I didn't give a lot of background on the VHL gene. The VHL gene is ubiquitously expressed. So it's expressed in every cell type. And so if you do a traditional just global knockout, the mice are embryonic lethal. So you have to utilize tissue-specific CRE to study the role of VHL in specific cells. What VHL is it encodes a protein that is a ubiquitin ligase. And when there is hypoxia in a cell, then the VHL will ubiquitin label the hypoxia. It will ubiquitinize the, sorry, I got that backwards. Sorry, it's late here for me. I know it's just morning there for you. But okay, so in normoxia, the VHL gene basically adds ubiquitin to the hypoxia inducible factor proteins, and so those are degraded. So in the presence of hypoxia, the VHL ligase is inhibited, and so the hypoxia inducible factors are stabilized, and then they can transcribe genes that are important for responding to the hypoxia.
Okay, so that's why you will have a future plan to do the tissue-specific knockout of the VHL, right? In the latter part.
Yes. Yeah, and part of the strategy too is that we first were very interested in osteocytes. Osteocytes are the mineralized bone cells that are embedded in the hard part of your bones. And the gene DMP1 was thought to be an osteocyte specific transcription factor. However, the DMP1 CRE mouse model has been shown to be leaky. And so it's expressed in other cell types, even outside the skeletal system. So we are, and it's not inducible. So we are looking to design more tissue-specific inducible knockouts, so that way we can really precisely answer our questions.
So actually, in relation to that, I have I have a personal question regarding. I have my own question regarding that part. So I was just thinking that the leaky blood vessels. So it means that they did not mature properly, and one of the players for the maturation of the blood vessels are the parasites. And from what I know is that the parasites also has a contribution in the maintenance of the stem cell niche in the bone marrow. So you also have checked some related studies on that.
So yes, you're correct. There is evidence in the literature that the parasites and the vasculature is also important in maintaining the niche. That's and particularly why I'm collaborating with Dr. Spencer, because his expertise is hypoxia and analysis of blood vessels, especially analysis of endothelial cells. And so we're not really sure exactly. We haven't done a parasite specific, knockout or but we're trying to get funding so we can use single cell RNA-seq to really dissect what is happening and changing in the individual cells of the bone marrow niche, especially rare cells like the parasites and changes in the endothelial cells in the bone marrow.
Okay, so we will look forward to reading that next paper. So again, for Miss Andrea, so how do we, there's a participant who asked, how do you disentangle metabolism from immune response? For example, whether the effect on the immune response is actually triggered from the metabolites due to the cytokine burst.
I don't know. I don't think. I mean, I think it's hard to disentangle, especially now. People are starting to look at immunometabolism, for example. But I mean, in terms of cytokines are pleiotropic. They don't just act on a specific way. They can act in different ways on cells. Maybe they can change a cell's metabolism, for example. So it's really hard to, I don't know, disentangle that. It's probably like a hand wavy cop out answer, but it's just something like it's a caveat. It's possible. And I'm not saying that the only, for example, when you have a cytokine response, cytokine responsive neuron, for example, the cytokine can act on the neuron, for example, in many ways. It can change its metabolism or synaptic property or something that they release. And we don't know how it's being done right now, but we're interested in looking into that further.
Okay, so may I add on that, because I think in your presentation you mentioned that the IL-4 receptor are also expressed in the neurons, right? And the cytokines directly modulate neurogenesis. That's one you mentioned. So I'm just wondering what aspect of neurogenesis is being affected.
So I think for that, in the paper that we recently published, we don't show that it's neurogenesis in particular. It's changing something in the synapse organization of the neurons. But in other works, people have suggested that astrocytes that receive the signal from the IL-4, they make BDNF, and that probably affects neurogenesis. It wasn't very specific how it was shown that they're affecting neurogenesis. It rescues some synaptic properties of the neurons. And I think it's probably also important to mention that I think adult neurogenesis is rarely seen. There are only certain parts of the brain that people have observed adult neurogenesis, like in the olfactory bulb and near the hippocampus. So I think we still don't know, but it's one of the ways that it can happen. Again, it's very hand wavy.
Yes.
I think this is like I don't know if it's a nascent field, but it's hard to study to isolate like this is just the only effect.
That's right. So there's still continuous development in this field, I suppose, because this is really one of the newest, probably. Is it? Is it one of the? Well, for me, it's new.
I think it's new in a sense because people have looked at the immune system and the nervous system relationship in the context of disease. So for example, when you have multiple sclerosis or Alzheimer's, neurodegeneration, that's how your immune system is implicated. But I think more and more we're starting to realize that there's some steady state healthy function of the immune system in the brain. And in a sense that's new. I think the paper that was published, one of the first papers that published how immune cells were affecting neurogenesis was in 2006 or something. I don't know if that's still new. It doesn't seem as new, but maybe it is.
Yes, thank you so much, Miss Andrea. And let's have just a last question for Dr. Manilay. So you briefly presented the relation or the interaction of sclerostin and inflammation in the bone marrow. So one of our participants is asking whether the NF-kappa beta pathway is a downstream target.
So I see. In the sclerostin knockout mice, I can't remember if NF-kappa B specifically was a target of the Wnt signaling that's disrupted through sclerostin, but I do know that our we did some RNA sequencing on the hematopoietic stem cells that reside in the sclerostin knockout microenvironment, and we noticed in those animals that there were a lot of gene sets that were related to experiencing inflammation, including osteoclastogenesis, which is activated through NF-kappa B pathways. And so we haven't done experiments to knock out the NF-kappa B pathways or look at, MIDI88 or any of those kind of regulators yet. That's something that we're hoping to get more funding to do so we can continue. But yeah, like Dr. Salvador's research, the interactions between the skeletal and immune system is osteoimmunology is also emerging, kind of new emerging field that's very complex because the changes are systemic. It's not just one cell does one thing and stays that way. And incidentally, there have been reports of the immune system and the neural system, regulating hematopoiesis in the bone marrow as well, both the peripheral and sympathetic nerves. And so it's really, it's hard to reduce the system to have, exact answers. And so we try to have exact answers, but we always know there's something else that has changed, and so we're trying to dissect these things one at a time and then see how they integrate. So I think it's going to be a while before we can understand at like the systemic level how things change, because things are also so dynamic.
That's right. So there are still many questions in our Slido app, but in the interest of time, I think we need to stop now. But because there are many who are interested, do you have any parting words to them before we end this session? We can start with Miss Andrea. You want to say something?
I guess before starting my PhD, I never really also handled the mouse brain. I didn't know where the brain. I mean, I've never seen a mouse brain, for example. And when I joined the lab, I was so green with anatomy, but I think it's a really emerging field, like neuroimmunology. Not just like how the immune system is affecting the brain, but really how the brain, for example, remembers immunity, and there are many other things that are unexplored. So maybe there are students or some young researchers there. Maybe it's something that you also want to pursue in the future because there's a lot of space. I mean, there's so many cytokines, there's so many behaviors. I mean, you can just do that your entire career, find the cytokine, find the behavior, find the mechanism, and I'm sure you won't be able to know the answer when you're done.
To just contribute, right?
But it's exciting, and maybe yeah, more Filipinos, like many more young researchers will be like interested in it in the future.
Okay, thank you, Andrea. And Dr. Manilay?
Yeah, so I would echo what Andrea said. So when I started my research as a graduate student, I was not even imagining that the experiments I was going to be doing, that I'm going to be doing bone marrow transplantation in mice, and what the implications were for that. Really, my questions and my curiosity is really for the basic science, but now that we know that there's some when there's some clinical relevance to it, it helps communicate the impact on the field. But I think as scientists, we are just curious and we ask questions and we try to design experiments, and we see something and we want to really understand it more. And I think that you should just try to always keep your curiosity and don't get discouraged when the funding doesn't come in, and try to and also try to collaborate, because the experiments that I'm doing now with my collaborators in different fields are really helping a lot to I think move the field faster. If we just stay in our kind of silos, we're missing out on a lot of information that can be integrated from other fields. And so I would encourage everyone, you have your specialty, but to, go to seminars and go to meetings like this, where you can hear a lot of science from, different perspectives, because it really stimulates a lot of thought and questions. And gets you to think more creatively about how you can approach your questions.
Thank you very much, two Filipino scientists that we have today. So we are very honored that you participated in our event. So I think we can go back with JV. JV, are you okay now?
Yes, ma'am. Hello po.
Okay.
So thank you very much po, Ma'am Larry, for facilitating our Q&A, and also to our speakers for sharing your expertise. And we would like to give you a certificate of appreciation. This certificate is given to Dr. Jennifer O. Manilay and Miss Andrea Salvador for the generous sharing of knowledge and expertise as resource speaker during session 4 of the Philippine Society for Developmental Biology 13th Annual Scientific Conference held online on November 17 to 20, 2021, signed by the PSDB Secretary Dr. Dino T. Tordesillas and PSDB President Larry Ann Ipulan-Colle. Thank you very much for sharing your expertise. We hope that we can invite you again. Thank you very much po. So now we're going to we're going to proceed sa photo opportunity po, and so kindly turn on your camera and wear your best smile. So sino po yung mag screen capture? Ma'am? Okay. Sino po yung pwedeng mag screen capture ng
Ako na lang po.
Okay, Sir Dino.
Okay, so we're still waiting for the others to turn on their cameras. Okay. There. So hopefully everyone can join us for this photo with our two speakers this morning. Okay. Mukhang wala nang kahabol. So I think we only have just one panel for this shot. So everyone, please smile. Look at your webcams and give us your best smile. So I'll be taking the photo in 3, 2, 1. Smile. Another photo, maybe a wacky one with some hand gesture. So 3, 2, 1. Smile. Thank you for then that's it. Thank you for everyone.
Okay na po?
Yes, sir. Okay na po.
Okay, thank you, Sir Dino. So, for the last 3 sessions, we presented the posters through video presentation, and today we're going to select 3 of the best poster presentations. But before we introduce the top 3 best posters, we would like to call on Dr. Amadresa Masaheri for the introduction of the judges and the mechanics of the poster live Q&A portion. Dr. Ahmad, good morning.
Good morning po. Saglit lang po. At mukhang nahirapan ang ating mga judges. The competition was a little steep. So, okay, let me just,
Okay, so before ma-introduce...
yeah, please kindly give me a few minutes. So,
Okay.
Okay.
Okay, so habang tinatali pa yung scores ng ating mga top 3 best posters, we would again like to thank the sponsors. First is Macare Medicals Incorporated, Merck, PhilAnaserve, and Medtest. Also, meron din po tayong photo contest. So, kailangan niyo po mag-take ng picture while watching PSDB 13 Annual Scientific Conference and use the hashtag PSDB13 or hashtag PSDB13ASC and tag us at PhilSocDevBio. And syempre, napapanood din po tayo via YouTube. So, kindly share the link dun sa mga hindi makapag-enter dito sa Zoom. And today, just a brief background na mangyayari ngayon. So again, we'll be going to select the 3 top best posters and they will be asked by our judges for questions. So kung hindi nyo pa na-visit yung mga poster, meron tayong link for the posters na sinubmit nila provided in the chat box. So yun. And also for those who are asking how to get a certificate of participation, again, first, you must be an official member of PSDB registered in the annual scientific conference. You must also submit at least 1 evaluation form in the evening session and the Saturday sessions. And take note that the evaluation forms will open at the end of the session until 11:59 p.m. Sir Ahmad, okay na po?
Yes, okay na po. Okay,
so
medyo nahirapan ang ating judges. Sige, I would like to say a few things muna. First of all, we thank the poster participants. We understand that it has been difficult really in these times to acquire scientific data and let alone make a poster. Pero nakakatuwa po na parang in this so meron pa rin mga nag-submit ng competitive na posters. So I first would like to explain the mechanics and then I would like to introduce our judges. So because of the pa-highlight na lang po host as I introduce the judges. So first, each poster participant was asked to submit a JPEG, and we decided to get creative this year by also allowing them to present a short clip, a short MP4 presentation of their work, which we played during the evening sessions. And we intended to play the top 3 today dahil po meron tayong tie just to how competitive it got. So 4 po yung aming ipi-play. So yung po ay na-forward ko na sa ating chat thread, PSDB board. But as they get prepared, so let me explain muna yung naging mechanics natin para alam natin. So there is a preliminary round, okay, where all the accepted abstracts were allowed to submit a poster entry in JPEG format and an accompanying video explanation for 4 or 5 minutes. And the judges in the preliminary round scored this one a total of 75%. That is 15% for the problem, another 15% for the specific objectives, another 15% for the research strategy, and the conclusion. Ayon. The other 25% po ay kukunin natin sa Q&A round in which initially the top 3 would compete but we have a top 4 kasi meron tayong nag-tie. So we will score them according to their knowledge of the research subject, which will get 10%, quality of answers to the question, which would be 10%, and composure and eloquence for another 5%, therefore completing 25% and total of 100%. So, uumpisahan ko na po i-introduce ang ating mga judges. I will give a brief introduction who they are. Although it would be least likely na we will appeals, but at least you know who were scoring you. The first one, the first in the panel of judges is Dr. Sonia Jacinto. Ma'am, can you open your camera so we can highlight you? Ayan. Dr. Sonia Jacinto really needs no introductions. She is one of the founding pillars of PSDB, and perhaps she is best known as the founder of the Mammalian Cell Culture Lab in the University of the Philippines. She also headed the Department of Developmental Biology for many years until her retirement. We still have her as one of our professorial lecturers. I could go all day long talking about Ma'am Sonia, but Ma'am Sonia, thank you for accepting this. It's really an honor to always have you. Yon.
Sure, Ahmad, anytime.
Okay, thank you po, Ma'am. Our second in the panel is Dr. Ryan De Chavez. Paki-hi, Sir Ryan, please. Ayan. Okay. Sir Ryan De Chavez is from UP Los Baños, a current faculty in the Institute of Biological Studies. And for those of you who don't know, ito po ang pinaka malaking biology department sa lahat sa buong UP system. He is also known as one of the curators in Natural Museum and very prolific malacologists. Yun, I could go all day long banggitin tong napakaraming publications ni Sir. Pero of those of you who have been interested in invertebrate zoology or mollusks na gano'n, so probably you have certainly heard of Sir Ryan De Chavez, and not to mention also served as a board member of the PSDB for 2 consecutive terms. So Sir Ryan, bumati po tayo. Nakamute po kayo, sir. Pa-unmute lang. Pa-unmute po, sir. Nako, hindi maka-unmute si Boss Ryan.
Wait lang po.
Okay, pa-unmute. Okay. Yan. Para naman marinig, Paki-unmute naman po. I'll next introduce
Ma'am Joyce, you're the host po. Can you please make...
Yeah.
Ayan, okay.
Can you hear me now?
Yes po.
Okay, so good morning to everyone. So thank you again for the opportunity to attend again after several, few missed conferences of PSDB. I really miss everyone here. So good luck to the presenters.
Yan. Sige po. Thank you, Sir Ryan. Always a pleasure having you. And of course, certainly not the least is also one of our faculty and one of our proteges na magmamana ng trono ng Department of Developmental Biology in the Institute of Biology UP Diliman. So the next judge I'm introducing is Ma'am Joyce Ferrer. Ma'am Joyce, can you please turn on your camera so that the participants in that? Yan. Ma'am Joyce is right now a faculty member of the Department of Biology, Institute of Biology, UP Diliman. But some of you may have known her already because she served, I think, 2 consecutive terms in the board of the Philippine Society of Developmental Biology. She's in her last term, she served as its PRO. So yung pong mga nakikita niyong magagandang pub mats last year dahil at souvenir invitations nung tayo po ay may face-to-face pa, siya po ang nag-spearhead nito. Kaya I had to invite this person. Her research interests include natural products and anti-cancer research. And ayun po, Yeah. Ma'am Joyce, again, thank you also for joining us. Sorry for our short notices. Yeah, Ma'am Joyce, bumati po
Honored,
tayo.
honored to be invited to judge the poster, sir.
Okay, sige po. Ayan.
And congratulations to PSDB for the successful proposal.
Okay, so thank you, Ma'am Joyce, and thank you sa ating judges who have one more thing to do, Judge. Ano? So I guess we are all eager to announce the winner. Dahil wet right out of the soup ito, babanggitin na lang po natin, okay, yung kanilang mga ano, and this will be in no particular order. Sige, babanggitin din natin yung kanilang scores and in no particular order. Okay, the first poster that enters our top 4, kasi we have ano, would be is authored by Maria Claret S. Suchia with the title Inexpensive Research at Home During Pandemic, Observing the Stages of Egg Development in Petmedeca Oryzias latipes. So ma'am, please get ready po to answer questions. This one has a preliminary score of 6.67. Sige, with the competition type. The next one, okay, sige Ma'am Lalet, please be on standby. Okay. The next one is authored by Miss Christine Nicole Padilla and the title is Optimization of the Protocol for the Transplantation of MCF7 Mammospheres on the duct chorioallantoic membrane. Okay, garnering a score of 66 points. Sige, wag na natin i-announce para ano. Okay. Okay, sige. And sige. And then next in our list of ano is okay, Miss Patricia Irene V. Trajano, effect of Garcinia binukaw crude leaf extract on skeletal muscle C2, C12 myoblast proliferation and differentiation. Yan. And certainly not the least, kasi ano, would be Mr. Jobriel Baluyot, detection of anti-androgenic activities in Laguna Lake surface water using a transcriptionally activated human cell. So congratulations to our top 4. To our top 4, please ready your cameras. Okay, and please be ready to answer the questions of the ano. So while we give you time to prepare, we will first play yung kanilang videos. Okay.
Okay, so habang pine-prepare pa ni Ma'am Joyce.
Good day, everyone.
Okay.
Good day, everyone. Today, I will be presenting our study entitled Detection of Anti-Androgenic Activities in Laguna Lake Surface Water using a Transcriptionally Activated Human Cell Line. A brief background. Laguna Lake,
Hello,
the largest
ma'am.
lake in the
Hello, Ma'am
Philippines,
Joyce.
is
Hindi po siya naka-full screen. Share screen po, ma'am, yung video mismo na naka-open.
For a while po.
Okay. So, yun nga, habang pine-prepare pa ni ma'am yung video, congratulations sa ating mga top 4 best posters. So, nakalagay na dun sa chat box yung mga poster o yung links for the poster. You can like their posters and then post comments and question for the authors as well. And also welcome po sa ating mga judges for today's poster Q&A. So yung pong dalawa ay naging judge nung ako po ay naglaban ng oral presentation nung 11 PSDB conference. But before that, before ulit tayo mag Q&A, gusto ko lang ipaalam din ulit na meron tayong photo contest. So just take a photo while watching PSDB and use hashtag PSDB13 sa Twitter or sa Facebook or hashtag PSDB13ASC. And we would also like to thank our sponsors in cooperation with Macare Medicals Incorporated, Merck, Phil Anaserv, and Meddes. Okay na po, Ma'am Joyce? Okay.
Good day, everyone. Today, I will be
okay na po, Ma'am.
in Laguna Lake surface water using a transcriptionally activated human cell line. Local water and fish in the Greater Manila area. However, the lake has become a basin for household, agricultural, and industrial runoffs, resulting in the accumulation of contaminants such as endocrine-disrupting chemicals that may affect the health of local fish populations and nearby residents. In fact, previous studies report EDC contaminants such as estradiol, progesterone, testosterone, bisphenol A, and per- and polyfluoroalkyl substances detected in the lake. To address this, we propose the use of human cell lines with transcriptionally activated luciferase reporter gene. Transcriptionally activated human cell lines with androgen receptors and estrogen receptors have been successful in determining androgen and estrogen disruption in water resources. These cell lines have high sensitivity to EDCs, are biologically relevant, and is high throughput. Here, we use human cell lines with a luciferase gene reporter that are transcriptionally activated by estrogen and testosterone receptors to screen for EDC activities in surface water samples of Laguna Lake. Our method starts with our collaborator conducting field sampling last March 2020, and the samples were run in solid phase extraction to concentrate the chemicals. The process samples were then tested using the cell lines and read using the luminometer for luciferase assay and spectrophotometer for protein to normalize luciferase activity. For the results of our study, our data shows that all water samples were primarily anti-androgenic and pro-estrogenic during co-treatment with estradiol. Previous studies report AR and ER agonism or antagonism by quantifying estradiol and dihydrotestosterone equivalent concentrations using a standard curve. We have simplified this approach by formulate an EDC index. Control, and one corresponds to US EPA threshold guideline levels. The responses for estradiol and dihydrotestosterone were then summed to determine cumulative response of each sample. Using our proposed EDC scoring index, sampling stations 4, 5, and 8 exhibit moderate endocrine disruption, while sampling stations 18 exhibit high endocrine disruptions. Furthermore, tributaries channeling to Laguna Lake were also found to contribute to high EDC signatures within the lake. Previous studies also report potential endocrine disruption within Laguna Lake, wherein one study found high vitellogenin levels with testicular degeneration among common carp captured near the East Bay sampling station, which is sampling station 18 in our study, compared to the reference group. Overall, this study provided evidence for the presence of biologically active anti-androgenic factors in Laguna Lake that may potentially inhibit male characteristics in fish and even in humans. This research is funded by the Department of Science and Technology, Philippine Council for Health Research and Development, And again, thank you all for listening, and this ends my presentation of our study.
So that's our first poster presentation. So from Mr. Jobriel Balino. Next presentation, ma'am, please.
I am Patricia Trajano and I will be presenting. Good day, I am Patricia Trajano and I will be presenting our research on the effect of Garcinia binucao crude leaf extracts on C2C12 myoblast proliferation and differentiation. Healthy skeletal muscles are vital for normal daily processes and high quality of life. However, extensive damage can be caused by physical activity, trauma, aging, and diseases, and can impair muscle function. Current treatments are not yet optimal to fully heal extensive muscle damage. However, Garcinia binucao leaves have been found to contain phytochemicals that have been proven to have muscle proliferation and differentiation capabilities. Thus, GB leaf extracts were tested for their effect on C2C12 myoblast cells to determine their pro-proliferation and pro-differentiation capabilities. To do this, methanolic GB leaf extracts were first prepared, while preliminary tests were conducted to visually determine the concentrations that exhibit effects on myoblast cells. The proliferation assay was done to determine if GB improves proliferation of C2C12 cells. This was observed by comparing the cell count of the treatments and controls in 24 and 48-hour intervals. An induction assay was also done to determine if GB can induce the differentiation of myoblasts into myotubes. The total myotube area for the treatments and controls were used to measure this. To visualize the cultures, they were first stained with a May-Grunwald stain and a Ginza stain, and the images were then obtained. To analyze the images, post-processing was done using Photoshop, and a Fiji method was especially developed for each of the assays. Specifically, a trainable Weka segmentation model and macro codes using thresholding were developed to properly analyze the necessary data for each of the assays. So here we see the results of the trainable Weka segmentation and thresholding procedures. In summary, for the proliferation assay 24 hours post-treatment, all GB concentrations exhibited a higher myoblast count compared to the gross medium control, with 0.195 micrograms per ml having the highest myoblast count. On the other hand, 48 hours post-treatment, all concentrations exhibited a lower myoblast count compared to the positive control. For the induction assay, 0.781 and 1.562 micrograms per ml had a higher total myotube area compared to the gross medium control. This indicates their potential induction of differentiation of the myoblast cells. Overall, the current data provides evidence for the potential dose-dependent muscle proliferation and differentiation capabilities of GB leaf extracts. From here, additional trials will be conducted to prove its statistical relevance, while an additional enhancement assay may also be done to determine if GB can further improve differentiation of myoblasts. So that is it for my presentation. Thank you so much for listening.
So that is Miss Patricia Trajano. Thank you very much, Patricia. Next presentation, ma'am, please. Thank you.
Christine Padilla, and I'm here to present my poster on my thesis entitled Optimization of the Protocol for the Transplantation of MCF7 mammospheres on the duct chorioalveolar alveolar membrane. So breast cancer is the most common type of cancer affecting women, accounting for the majority of cancer deaths worldwide. Over the years, there have been tremendous developments in the detection and treatment of this disease, and for several decades, the MCF7 breast adenocarcinoma cell line has been the most extensively used both in vitro and in vivo in cancer biology studies. Despite the significant use of both approach, however, in vivo studies possess several advantages, and one of which is the capacity to observe the cancer under conditions resembling the natural microenvironment. The golden standard murine models are widely being used for tumor transplantation, metastasis detection, and anti-cancer activity assessments. With its increasing contribution to cancer research, however, come its high costs and, more importantly, the significant number of sacrificed animals. Therefore, it is invaluable at this point to develop alternative models which allow the conduct of the same, yet more humane animal studies. So here we present the chick embryo chorioallantoic membrane model, which takes advantage of the vascular network of this extraembryonic membrane. As a matter of fact, this cell line has already been proven to form tumors on the chick cat. Note, however, that considering the higher availability of fertilized duck eggs as compared to fertilized chicken eggs here in the Philippines, we have decided to adapt the model to the former instead. So our results can be divided into 4 parts. So here we can see that we determined the human cancer cells and cancer types viable for in vivo implantation. Next, we then check the applicability of the duck egg to the transplantation of MCF7 cancer cells using 2 setups. So the first one involved a negative control group treated with PSP and an experimental group inoculated with MCF7-derived mammospheres. Here we have observed the emergence of histological alterations, namely cell infiltrations and dense mesenchyme, which were both suggestive of a possible tumor formation. For the second setup, we now decided to introduce MCF7 cancer cell suspension in the negative control and MCF7-derived mammospheres in the experimental group. So here we have seen the emergence of tumor nests, which are comparable to the ones observed in the study of Roberti on pancreatic cancer cells. The third part of the study involved the markers which can possibly be used to detect the cancer cells transplanted on the cow. And lastly, we also present the uses, advantages, and disadvantages of the model. Thank you.
And for our last poster presentation,
scientific research, remote learning setup, research at home. In light of the global pandemic, we need to think outside the school laboratory that is inaccessible when approaching the research. Where would we begin if we want to be inspired and experience a remote scientific activity that is relatable and accessible? Available at home setup such as our little bowl of aquarium at the corner of our desks, intended as a hobby or for entertainment, may provide a fun learning scientific activity. In our poster, we present our embryology at home, a chip and a simple setup to observe the different stages of development in the Medaka fish. The Medaka fish laid eggs every day from mid-February to August 2021, and eggs were put in used plastic cups and plastic box containers available at home. Then images and videos of the different stages of egg development were captured using a 1,700 peso mini pocket microscope that can be connected to mobile phones, tablets, and laptops. With this, we were able to observe the different stages of egg development in Medaka, from activated the egg stage, blasted disc formation, 2-cell stage, 4-cell stage, 8-cell stage, 32-cell stage, morula, blastula, gastrula, organ formation, and until the hatching stage. Everyday observation of this natural phenomenon and data collection provided a quality learning experience and at-home observation of a direct key in animal development. We hope that our poster will inspire students to develop foundational research skills such as data collection, synthesis, and ability to report findings. For teachers like me, we hope that our poster would inspire you to upgrade your lesson materials with original content based from the pictures and videos from this inexpensive digital microscope. Further, we hope that you can also do this at the comfort of your home and to share the joy of science among family members.
so Thank you very much to our top 4 poster presentations. Again, congratulations for making it this far. And we're not finished yet because we'll be having our poster live Q&A. But before that, Mr. Ahmad has a announcement. Sir?
Yes, thank you very much, JV. Okay, so those are the top 4. Okay, we will be ranking first, second, and third depending on how well they will be able to answer the questions of our judges. So to our judges, each you may ask you may ask 1 to 2 questions to each of our top 4 finalists, and each contestant, for each contestant, we will allot 5 minutes so we don't drag this. Later on, the judges and I will have a breakaway group and we will compute everything and we will announce the winner this afternoon. So wag po kayo mawawala ngayong hapon. Sige, I will randomly select na lang, okay? Mr. Jobriel Baluyot. Okay, please turn on your camera and please unmute yourself. Okay, spotlight po si Mr. Baluyot. And Mr. Baluyot, if you are ready, please let us know. Hindi pa yata can we give him co-host privilege? Okay, sige, para rin tayo.
Co-host na po si Mr.
Ayan, okay. Teka lang. After Mr. Jobriel Baluyot, we will have Ma'am Suchia. Okay, Ma'am Lalet Suchia, followed by Miss Padilla and Miss Trajano. So yun po yung order natin. Okay, Mr. Baluyot, are you ready to answer some questions?
Yes, sir.
Okay, sige. Okay, Mr. Baluyot has his 5 minutes. So sino pong unang judge ang magtatanong?
Okay, so
I'm
hello,
sorry,
Joriel.
Yes. Good morning,
Okay.
sir.
So congratulations on your excellent poster. So I have 2 questions for you. The first one, did you specify or did your study specify the particular androgenic or estrogenic chemicals that you've studied here?
Actually, the way that we measure the androgenic and estrogenic, so the short answer is no, because the way the method works is that any chemical that has an affinity to the receptors, be it the estrogen receptor or the androgen receptor, will bind to these receptors, and then eventually that will result in a signal. So what we were generally measuring is an array of chemicals that would potentially be interacting with these receptors. So we were not able to identify what particular chemicals are interacting.
Okay.
But that may be a future approach towards the method.
Okay, so my second question is, what do you think, aside from the hydrologic flow that you presented in your poster, what are the other possible factors that might explain the EDC variability that you observe in the different areas of Laguna de Bay?
Okay, thank you. For the next question, I think one of the other factor that we were interested into looking to is the presence of industrialized systems in that particular area. So that area is where I think most of industrialized or industries in Region 4A is located. And in fact, one particular study by Velarde and his colleagues has found that individuals, especially women that are situated near that area, is actually presenting high levels of endocrine disrupting chemicals in their blood. So I think that is one of our speculations for the moment on why there's a particular increase in the endocrine disruption around that area.
Okay, Mr.
Okay.
Baluyot has 2 more minutes to be questioned. Okay, I think Ma'am Joyce, kayo po ang naka-highlight.
Morning, Ma'am Joyce.
Good morning, Job. So my question for you is what's next for this study? So what are the next steps so that this can eventually lead to policy recommendation?
Okay, thank you. For that question, actually, we are pursuing some form of policy paper to actually move the technology forward. And we're hoping that this can be used to actually assess and regulate drinking water at some point, because the method is not really something that is currently available in the Philippines and in the US. Actually, in the US EPA, the US EPA is utilizing this method to regulate drinking water in their system. So I think it's something that we can adapt here in the Philippines in the near future.
Okay.
Sorry. So, but
So for
what
the
about
experiments,
that's for the technology addressing the quality, like
yes,
the quality of drinking water from those sources. But what about policy? Because you have you were able to trace the you were able to rate the level of contaminants from the different tributaries or from the different sites around the lake. So what about for that aspect? I think this is a bit related to the second question of Sir Ryan. So yeah, so you have any recommendations
okay.
for that?
So for that aspect, I think the more appropriate method to support this experiment would be the use of targeted analysis, like for example, LCMS, to determine what particular compounds are responsible, and so that we can trace back what is causing the increased anti-androgenic or pro-estrogenic responses that we are observing within the lake. And yeah, I think that will be the more important questions that need to be addressed, because as of the moment, we only know that there is a response from the lake, but we don't know what is causing the particular response.
Okay,
Thank you.
Mr. Baluyot, we can excuse Mr. Baluyot. He has used up his time. So thank you very much, Mr. Baluyot, and good luck to you.
Thank you very much.
So may we call on now Ma'am Ma'am Claret Suchia. Okay, can we please highlight her? Okay, thank you very much. Okay, Ma'am Lalet has her 5 minutes. Who wants to ask the first question? Ma'am Sonia, I think you're muted.
Lalet, hello.
Good morning, Mom.
Yeah, that's also a very good poster there. May I just ask if ito bang medaka na ito ay madaling ma-procure dito sa Pilipinas? Every time I search for it, ano ba yung lumabas sa akin? Ginshin Impact something farm. Oh, parang Japan ano siya. But yeah, can we buy that locally? And is it very prolific as well in the Philippine setting dahil we are problematic with our Danio breeding.
Right. Thank you very much, Dr. Jacinto. The setup was in Japan. So the Medaka is a Japanese breed. In the Philippines, locally, we have Danio Ririo, and the size is the same as that of the Danio Ririo, and the color, there is a white, the yellow, and the orange one. So it can be likened to the zebrafish, which can be purchased locally. And the price of the Danio Ririo, as you know, if you can purchase locally in a pet shop, is 20 pesos to 25 pesos per fish. And regarding to its reproduction, its fecundity, yes, there are certain months that you can collect the eggs of Danio rerio, same as that of the Medaka. So I've researched about it. Speaking about Medaka, there is a species, Oryzis luzonensis, that we need to study. Hence, this made me also inspired that when I go back to the north, because I'm from Isabela and to the Ilocos, so that was the last reported case or where they found the Luzonon, the Medaka, the Medaka that is very endemic in the Philippines. So that would be probably my next step, that I would really want to rediscover the Philippine Medaka.
Okay, so second question, last na lang. For this type of activity, you really have, or each student will have in his or her home, yung sabi mo, microscope. No?
Yes, ma'am. I'm ready. I can show you. This is the microscope that I use. It's just at the palm of your hand. And I'm ready with, in Lazada, you can purchase at even cheaper than what I have purchased. It's 576 to 800 pesos in Philippine peso, ma'am. You can just type in Lazada microscope, and there you have.
Yeah. Okay. All right. So give way to the other judges.
Thank you very
questions. Thank you, Lalit.
So Ma'am Lalet has 1.5 minutes. Just to remind it, ano lang. So we keep the competition fair. Sige. I think the next one who will ask questions is Sir Ryan. Naka-mute yata ulit Sir Ryan. Yes, he is. Kindly unmute Sir Ryan.
Okay. Hello, Lalet. So my question is, in using Danio rerio as a model organism for laboratory studies, one of the advantages is you can induce it to lay eggs. Okay, but in the case of the case of Medaka, can you also use, or what are the do you also have a protocol in induction of the eggs so that you can easily observe that in, for example, you want to collect and study the eggs for different experiments?
Right. In Japanese Medaka, we have observed, because it was originally intended as pets. We didn't really expect that we would collect eggs from February to until the end of August. So that would mean about 6 months that almost every day they can lay eggs. So I think probably I need to look more into that, but I haven't found induction per se, but it's more of the temperature. If the temperature is above 20 degrees centigrade, then that would be most feasible environment for the Medaka to mate and to produce eggs the following day. That was our observation so far for the 6 months. So it's all natural that they lay eggs almost every day.
Okay, thank you very much. Ma'am Lalet has used up her time. We will move to the next contestant. Good luck to you, Ma'am Lalet. Okay, Miss Padilla, are you ready? Okay, can you please turn on your microphone and unmute yourself? Okay, can we please spotlight Miss Padilla? Okay, there she is. Okay, Miss Christine Nicole Padilla has her 5 minutes. So, okay, sino pong mauuna magtanong? Si Ma'am Joyce siguro naman.
Hi, Christine. So I'm interested in the setup where you compared introduction of mammospheres versus cell suspension, because in mice they usually use a high cell density cell suspension to induce tumors. So what happened in your setup for the cell suspension as compared to the mammosphere?
According to the results po, only the setup involving the introduction of mammospheres were able to form histological features that suggest tumor formation. So we weren't able to observe that when we used only the cell suspension.
Okay, sige. Sino pong gusto magtanong, sinong judge po yung gusto magtanong na susunod? Or does mom judge? Yeah, Sir Ryan, go ahead. Okay, pa-unmute ulit kay Sir Ryan.
Okay. So congratulations, Christine, for that excellent poster. My question is, do you know a possible mechanism, if ever, on the establishment of MCF7 in CAM or CAM?
One of the papers that I was able to read in the past was the ability of MCF7 to actually make use of the mesoderm of the CAM as its surrogate tumor location. Basically, it induces the mesenchymal stem cells around it and turn it into vasculogenic progenitors, which allow it to have new access to the blood vessels around it and basically make use of them to acquire nutrients from its environment, contributing to its tumorigenic capacity on that kind of rich vascular network.
Okay, so can I have a follow-up question?
Yes,
okay, so my follow-up question is related to Joy. So are there, do you have any recommendations for the further improvement of your protocol using MCF7 in CAM?
Since we weren't able to actually finish the protocol due to unforeseen circumstances, I can recommend using tumor markers so that we can actually confirm the presence of tumors using the platform, since we just stopped at the tumor formation stage. We weren't able to confirm it.
Okay, thank you.
Okay, she still has 1.5 minutes. We can ask her one more question. Okay, Ma'am Sonia.
Can I ask a question?
Yes, Ma'am Sonia.
Okay. Yeah, hi Christine. I have encountered na many times your poster, but anyway, your work. Yeah, okay. So one, you showed some cells that you think were tumor nests or clusters coming from the MCF7, right?
Yes po.
But hindi pa mo pa talaga na-establish kung totoo nga siyang tumor cells coming from the growth of the MCF7, right?
Yes po.
Now, whether it is really tumor cells from MCF7 or just cells, kagaya ng sinabi kanina, na mesenchymal cells ng na nag-proliferate lamang. Tama?
Yes.
So, ito yung mga next steps. Gusto ko lang ma-clarify yan. Okay, salamat.
Okay, so right on the bottom, I think, yes, Miss Padilla's time has run out. Thank you. We will excuse you, Miss Padilla, and good luck later. Okay, stay. Okay, certainly not the least. Okay, our next Miss Trajano, we would like you to turn on your camera, please. And can we spotlight her? I think I saw she was already here kanina. Miss Patricia Trajano, can you hear us? I see you are in the Okay, Miss Patricia Trajano.
Ayan.
Ayan. Yeah. Sige. Okay, can we please have her unmute herself? Okay, she is already in the spotlight. Hold on. So that it's a fair game. Let me time that. Okay. Miss Patricia Trajano now has her 5 minutes. So who wants to ask the first question? Yes, Sir Ryan.
Okay. So congratulations, Patricia, on your excellent poster. So my question is, in your result, you have two concentrations in your induction assay variability that would have higher levels than the test setup of GM. So what do you think is the reason behind such variability compared to other non-GM exposed tests?
I think those two concentrations po represent the dosage that would be enough to induce the differentiation of myoblasts into myotubes. And then I think there are only two concentrations, and the other concentrations that are more than this would be in a way toxic to the cells, which is why we observed a significantly lower differentiation than the control.
Okay, so I have a follow-up question. Now, since you're talking about the ability of Garcinia in muscle proliferation, so have you read about, or do you have any hypothesis in the possible mechanism of action of the possible active components of Garcinia in skeletal muscle proliferation?
So these chemicals, such as polyphenols and flavonoids, they have been seen to either act as an antioxidant to the muscle cells, which is why this promotes differentiation and proliferation or proliferation. And another possible mechanism would be the upregulation of myogenic factors such as myogenin and MS4.
Okay, thank you.
Okay, so, Ma'am Joyce, I think we'll ask the next set of questions.
I also have a question for Trish related to the first question of Sir Ryan. So in your differentiation assay, it was 7 days long treatment, correct?
Yes.
Is that correct? Okay. So I'm curious if so you identified, I think, two concentrations where the differentiation was greater compared to the control. But for the other mostly higher concentrations, did this cause death of the muscle cells?
No.
I don't think we can determine po if they cause death, but rather maybe they just inhibited the differentiation.
So you only observe less differentiated cells, but overall, it was not notable that there was a decrease in the overall amount of cells, including the stem cells naman. Is that right?
Yes, ba. They just our way of measuring the differentiation po was through the area of the cells, and the cells were still there naman po, but they just didn't increase in area.
Oh, okay. Thank you.
Thank you. She still has a minute to answer one more question. Okay, Dr. Jacinto, please go ahead.
Okay. Good morning, Patricia. I like your poster and your results, but I just have one on the research design. Parang ako doon sa, I think, exception to the what is positive control that you use. Kasi ang positive control mo yata is digest the growth medium, tama? Tama ba?
Yes, ba.
And then you have in your experimental setup, so this is an experimental setup, growth medium din, tapos naglagay ka ng dinukaw na extracts. What I know about positive control is that this should be something na alam mo talaga na it will really get that effect that you wanted. So it could have been polyphenols, no? And then now you said na antioxidants, ang polyphenols, that could be the reason why, so you could have used vitamin C or what. Yun lang ang that's really kind of comment, so that if you want to improve your research, you can do that. No? Okay.
Yeah, I think, right on the button again. So Ayun, her time has already been up. So thank you very much, Ms. Trajano, and I have to say magagaling din po ang ating mga judges. So I'm so glad you said yes po to this. So ayun, sana di po kayo magano sa mga invitation ng PSDB. Okay, thank you very much. Okay. So from here, the judges will have a breakaway session. I will communicate with the judges through our FB Messenger na lang. And I now turn over the floor again to our Master of Ceremonies, si Mr. JB Nabalan. Okay, thank you very much.
Thank you so much, Sir Ahmad, and then to our judges po, and again to the authors of the poster presentation. Before we proceed to the photo contest winner, I would like to inform everybody that we will be having our afternoon session at 1 p.m. and doon na po natin i-announce yung winner ng ating best poster. And syempre, sabi nga natin kanina, we have a photo contest. So, nasa kamay na ni Ma'am Larry ang winner. So, Ma'am Larry, can you please share the winner of the photo contest? So, the winner is, hindi ko makita yung username, pero siya yan with caption, Saturdays like stem cell, unique and specialized. So congratulations, kokontakin ka ni Sir Judel for more information and updates kung paano makukuha yung prize. So congratulations. So again, thank you very much for attending this session 4. And mamayang hapon, we will be having our session 5, talking about developmental biology for conservation with Dr. Pavia and Dr. Anticamera, and then awarding ceremony of the best posters. And again, thank you very much for attending this session. Mabuhay po tayong lahat. See you and please be back at 1 p.m. for session 5. Thank you po. Advance happy birthday, Miss Karen Davila. November 21 na bukas. Thank you so much po. Mabuhay po. Ingat po tayong lahat.
Hello po everyone. So we will be back po ng siguro kung hindi po kayo confident with your Zoom hosting or gusto niyo po mag-practice, you can, hindi ko na ito papatayin. Bala na po kayo mag-practice with your members. If gusto niyo pong mag-practice.
Pero i-stop natin yung yung streaming sa YouTube.
Okay po. Si Ma'am Joyce, can you please give me back the pala. Wait. I can reclaim it.
For a while there.